Well, welcome back from spring break. I hope you were able to enjoy some time off. Or if you were like me, you perhaps got a lot done. So, either way, I hope that it was useful. Um, all right, so we're going to begin this week, and we're going to talk about cells. And before we do that, just a little tribute to a blues singer. This is Jimmy Rogers, and he died at the age of 35 of tuberculosis. And he wrote a song and performs this song called the TB Blues, and you can listen to it and watch it, I guess, on YouTube. It's rather sad. But it's fascinating. It's definitely a part of our history. There was a time when there was a good chance you wouldn't live past middle age because of tuberculosis. It was the number one killer of human beings. Thank goodness for antibiotics. Hopefully they hang in there, right? All right, so we're going to talk about cells. Really, microbiology is cell biology. So, this is a very important foundation for the rest of this particular course. So, we're going to break it into parts one and two. I just do that just to make sure that you take a break somewhere in there. So, all right, let's begin. These are the objectives for the week. I have a particular objective that I wanted to point out, compare and contrast gram positives, gram negatives, acid fasts, and atypical bacterial cells. So this is rather important. And then this week's assignment has to do with describing a virulence factor that is related to your pathogen. And if you're wondering, what is a virulence factor? Okay, well, we will get there. Actually, that has to do with the next objective down the line, to be honest. So we will get there. All right, and here are some terms as always. There they are. Excuse me. I have a few terms that are particularly stressed and highlighted in red. We're also going to talk about some various bacteria, the poster children that relate to certain topics that we are going to cover. So make sure that you know these organisms as I describe them in the lecture. All right. So compare and contrast prokaryotic and eukaryotic cells. Let's just do a brief comparison. Years ago, or maybe just a while ago, you probably learned this and maybe promptly forgot it. At least, and maybe you didn't learn very much about something called prokaryotic cells, but eukaryotic cells, you should have had that at some point. And I'll explain what that is in just a moment. But a cell, cells. Why are we talking about cells? What are cells? So, all right, cells, if you remember way back in biology class, cells are considered the smallest unit of life. Now, that was just something that I memorized, but what does that really mean, especially as it relates to microbiology? Well, understand if you're talking about a living thing, it has to be a cell or many cells working together to make that living thing, that living organism. All right, so living things are cellular. All right. And some living things consist of just a single cell. That would be like our bacteria. And some things are multicellular. And that means that, okay, they are made up of many different cells that hopefully get along and work together. Of course, we are multicellular. We are example of that. And if you're wondering, okay, why is she really going over this? This is kind of a biology part of the class, and isn't everything cellular and living, and all these microbes and infectious organisms, aren't they all this way? No, actually, we'll talk about the particle that causes mad cow disease, which is infectious, and we talk about it in microbiology, but it is not living, it is not a cell. Also, we will talk about viruses. Viruses are technically not considered living. They're really not cellular, although there's a weird argument for that that we will talk about a little bit later on. All right. So, all right. So, living things can be a single celled thing, they could be multicellular things. Okay, what's this prokaryotic, eukaryotic thing? All right. So, there are two types of cells on this planet. The very primitive types of cells have not evolved a nucleus. So, these are called prokaryotic. Pro always means before. Okay. Kary, when you see Kary, that's always talking about a nucleus, all right? So pronucleus, prenucleus. Okay, so these are single-celled creatures that they didn't develop and evolve a true nucleus yet, okay? There are lots of creatures like this, and this would include your bacteria. Okay, there are also eukaryotic creatures now. And oh, one other thing, sorry, let me backtrack just a minute. Prokaryotic creatures are considered single celled. Okay, they're not really multicellular prokaryotic creatures. They are technically considered single celled and they don't have a nucleus. Now there are exceptions to the rule. There always are in biology, and that is actually very important part of biology. Everything sort of it develops into and evolves into. So there has to be intermediates. There's always those exceptions, and that is true also with this. Interestingly enough, these single-celled creatures talk and communicate with one another, and in the body, the prokaryotes that live in and on your body talk to one another and even gang up on you. So there you are, almost like a multicellular creature. All right, sorry. Now moving to the next type of cell. These are eukaryotic cells. Now, eukaryotic, EU always means true. So true, and then remember K-A-R-Y, that means, okay, nucleus, true nucleus. Ah, okay, so these types of cells have a true nucleus. Prokaryotic, they don't have a true nucleus. Eukaryotic, they have a true nucleus. Got it. Now, eukaryotes are all multicellular? No, actually, most eukaryotes are actually single-celled and invisible to the naked eye. But of course, the ones we tend to be interested in are the multicellular eukaryotic creatures, and they are interesting. That would be us and your dogs and your cats and your horses and all those the birds and the plants. They are multicellular and they are eukaryotic, and they are the most interesting, clearly, but because they're so complex. All right. So again, eukaryotes can either be single or multicellular. All right. What's a good example of a eukaryotic single-celled creature? Amoeba. All right, that's just a real fast one off the top of my head, but there are many other kinds of yeast, and we'll talk about a few of these. All right, so if we're going to measure a cell, what kind of unit should we use? Well, microbiology is the study of cells. Microbiology, okay, if we're going to measure, maybe we can use a micron, or also known as a micrometer. Okay, microbiology studies cells and we use microns or micrometers. Now, when designating a micron or micrometer, of course, we have the M for meter, and then we have this little symbol here, which is the Greek letter mu. This is for micro. All right. So now, if you're wondering just how big a micrometer would be, we will talk about this a little bit later on. But if you could take a quarter and divide it into 1,000 even slices, boy, that would take a sharp knife. If you could cut a quarter into 1,000 slices, thin slices, one of those slices would be a micrometer. Okay. So it's pretty small. Okay. And okay, you can see the width of a quarter, that's about a millimeter. But if you sliced it into 1,000 different slices, one little slice is about a micrometer. Yes, give or take. All right. So let's do a little comparison between those prokaryotic cells and eukaryotic cells beyond the fact that prokaryotic cells don't have a nucleus yet and eukaryotic cells do. So, all right, prokaryote, as I said, pre-nucleus. What's an example? Ooh, bacteria. Raise your hand if you know of any bacteria. All right. Hopefully, you realize that the creatures that you are doing your report on are bacteria. Okay. General characteristics here, and I have a little illustration. They're very simple, as you can see. They're really small. Now, small is a relative term, but typically the bacteria that we tend to study in microbiology are somewhere around 3 micrometers. It depends. I know one of you has a fairly long one, but typically they're, somewhere between 10 and 2 micrometers, just on average, but there are some that are much smaller than that and some that are much bigger than that. Okay, they are very simple. All right, well, they haven't evolved even a real nucleus yet. So, yeah, small and simple. Okay, and again, they are always single celled or unicellular. They are single celled living creatures. They have ribosomes, and if you remember, okay, ribosomes are really important if you're going to make proteins. And let me tell you, every cell really, really, really wants to make proteins. Okay. So every cell really needs a ribosome and they really need that ribosome working well. So ribosomes make proteins. They assemble amino acids, right? If you remember that, right, from Mr. Seberg's lecture. All right. Well, prokaryotic cells have very small little ribosomes. They're considered 70S ribosomes. Now, the S stands for Svedberg. No, don't write that down. Svedberg is just an old-fashioned way to measure particles, and they used to spin things down in a centrifuge and measure how far they went and blah blah blah. If you really want to know about Svedberg units, look it up on Google. But it's not really all that important here. Now, they don't have a true nucleus, but there is a place for their DNA chromosome, and this is in a nucleoid area. So something that isn't real or true, sometimes we say it's like oid, like humanoid isn't quite human. Nucleoid is a nuclear like area, but it's not a nucleus. Okay, so not a true nucleus. Now, prokaryotes only have a single circular chromosome. They must always have that single circular chromosome. Do they have more chromosomes? No, never. If they lose the one, will they survive? No, they will not. Now, they might have these other structures that you see here in blue. These are called plasmids. Can they live without plasmids? Technically, yes. Okay. Plasmids are extra little pieces of DNA. They're also circular, and they would fool you. They would make you think that this is a chromosome because they're real DNA structures. It's a double helix, just like all DNA, but they're small and they carry extra pieces of DNA. They might have, for example, genes to make toxins here. Okay, and they might have genes to make special structures or genes to become antibiotic resistance. And plasmids can be shared between one bacterial cell and another. All right, because these bacteria are so simple, they really don't have a lot of stuff. Okay, technical term stuff. If you don't have a lot of stuff, you really don't need to put things into compartments, and that takes away from the efficiency of the cell. If you have blockades and like what we call membrane enclosed organelles, that would actually take up resources and block the movement of molecules. So these are simple, they don't have a lot of stuff. They do not have membrane enclosed organelles. Now, if you're wondering, now what is an organelle? I do remember that term, but I've forgotten the definition. An organelle is a structure in a cell with a function. So you could almost say an organelle is a cell thingy, but it's something that a cell has. It has a particular kind of function. So the prokaryotic cells do have organelles, but they don't have membrane enclosed organelles. Okay, they're not enclosed in a membrane. They're not enclosed in compartments. Now, if you have a lot of stuff, like a eukaryotic cells, like a eukaryotic cell, then you need compartments. How would you make those compartments? You would put them inside membrane, little sort of packets, compartments, and that's what you would do for eukaryotic cells. So, all right, let's move to a eukaryotic cell. Now, I've shrunk down. This shows a much smaller kind of green bacterial cell, but you can see the comparison. Here's a bacterial prokaryotic cell compared to one of ours. And this is one of my favorite kinds of eukaryotic cells. This is a white blood cell. And what is it doing? It's actually eating bacteria. This is one of our soldiers. This is a macrophage. And the macrophage goes in and out of tissue. It goes looking for bacteria like this, which are prokaryotic, and it grabs onto them and it pulls them in and it digests them. So you can see that, oh, yeah, this is a big complex cell and it's going to have a lot of stuff, and that stuff is going to have to be placed into membrane compartments or membrane organelles. Okay, there's big factories that are going to manufacture things, break stuff down, and they also have to be in membranes. All right, so this is a eukaryotic cell. Eukaryote means it has a true nucleus, okay? And here we have our nucleus, or excuse me, here's our nucleus here, and it was showing a nucleolus. That is different. That makes ribosomes, if you forgot what that is. But this whole structure here, it's membrane enclosed. It's a true nucleus. That makes our white blood cells a true eukaryotic organism, okay? All right, so examples of eukaryotes. Well, again, human cells are our favorites. Our white blood cells are amazing, like our white blood cell here, which is a macrophage and it eats bacteria, so we love it very much. We're going to talk about our soldiers, our white blood cells, but we also will talk about fungal organisms. Now, the reason we talk about fungal organisms in this class is because they cause fungal diseases. And it turns out these fungal organisms that cause disease are very different than the bacteria that cause disease. How different? They are eukaryotic. They have a true nucleus. Okay. Now, if you're wondering, okay, that's fine. Do I really care? Well, it will become very important to understand that these are eukaryotic when we talk about killing them with poisons that we call drugs, okay? All right, the protozoans, there's a lot of protozoans that cause disease out there, and they also are eukaryotic. This would include amoeba, like the brain-eating amoeba, flagellates, the ones that cause, oh gosh, giardiasis and trick. There are ciliates out there and something called apicomplexans. This would be like the organism that causes malaria. Okay, these are all protozoans and they are eukaryotic. So, what are some general characteristics of eukaryotic organisms? Well, they have a true nucleus, a membrane enclosed organelle, and they are much larger. And this is, I didn't have room to make this cell larger, so it's not to scale. But okay, again, eukaryotes can be unicellular, like yeast, or multicellular, like us. They are very complex. Oh dear, yes, they are, but they're cool in that way. Now, their ribosomes are 80S. Okay, look over here. Remember the ribosomes over here were 70S? Okay, over here. The ribosomes are bigger in a eukaryotic cell. So they're different. Why do we care about that? Well, we can maybe send in a poison to kill bacteria that only hurts 70S ribosomes. Okay? Or maybe hurts them more. So again, it has a true nucleus. Now, the chromosomes that eukaryotic organisms have tend to have, they are linear, so they're not circular. They're not circular, they're linear, like a line. And usually eukaryotic cells have many of them. Replication tends to be extremely complex because of our many membrane enclosed organelles, also because of our many linear chromosomes. Oh, golly. So those have to be replicated, and we have to go through a nuclear division before the cell can divide. And if you'll remember from your basic biology, okay, so, nuclear division includes mitosis or meiosis, and then the rest of the cell can divide up, and that's cytokinesis. We're not going to really go through that in this class, but understand it is more complex, and there's some relevance to this too coming up. And again, eukaryotic cells, do they have organelles? They do too, except that their organelles are a lot of them are membrane enclosed because they've got a lot of stuff and they've got factories and they've got to put things into compartments. And this is the reason I love this illustration here, because this macrophage, our white blood cell that's been patrolling the area, has grabbed the bacteria and brought it into a little package here, a membrane enclosed package, and it's going to send some digestive enzymes over to digest it. And these are pretty deadly enzymes, so they have to be kept in a membrane package. And these are called lysosome packages, by the way. All right, this is a little larger so you can see how this all transpires. You have the bacteria being grabbed, engulfed, um, digested by the fusion of a lysosome to this membrane holding the bacteria called a phagosome. And the bacteria get all digested, so it's kind of cool. But again, very simple over here, no true nucleus, maybe a nucleoid area. Over here, okay, a true nucleus, very complex. Okay, true nucleus. Okay, you got this. Prokaryote, eukaryote. And all right, so what is this? This is a macrophage. And he's sending out these little long, what are called pseudopod-like structures, part of the membrane, and he's grabbing the bacteria and bringing them in. Kind of cool. And you can see the difference in size, and you can see the complexity of a eukaryotic cell. I know it's a little bit busy, but here's the outside of a macrophage, and they send out these long, long, like fake, well, they're called pseudopods, fake feet, but they're almost like arms, and they grab the bacteria and bring them into these little vacuoles to eat them. Okay. All right, here's another illustration. This is another type of soldier, one of our white blood cells. This is called a neutrophil. And oh, look, it has a true nucleus in there. Now, this guy has grabbed some other kinds of bacteria. These bacteria are the kinds that cause Rocky Mountain spotted fever, and they are stained bright pink. And now this is again a way to be able to compare size and complexity. All right. And again, this is Rickettsia rickettsii that causes Rocky Mountain spotted fever in bright pink. Notice the size difference. The white blood cell is actually stained blue. Okay, our neutrophil white blood cell is stained blue. Much more complex, has a true nucleus there. Okay. They have membrane enclosed organelles, although it's rather difficult to make out in this illustration, I know. And it's hard to see the bacteria, they're so small, and there's no way to be able to tell for sure that there's no nucleus. In fact, there's a nucleoid area, but you're just going to have to take scientists' word for this one. So again, red bacteria, small, very simple, prokaryotic, no true nucleus, not visible in this picture. Now, again, both cells have organelles, but only the eukaryotic cells have membrane enclosed organelles. All right. Which cell type has a true nucleus? Prokaryotic, eukaryotic, both or neither? You got it. B, eukaryotic cells. Very good. Which type of cell has organelles? Prokaryotic, eukaryotic, both or neither? Now, did you say both? Yes, an organelle is a structure in a cell that has a function. Prokaryotes have organelles, but only eukaryotes have membrane enclosed organelles. Okay? I know, sometimes that one is very confusing, and I've heard instructors not quite getting this right too. All right, so which type of cell has DNA? Prokaryotes, eukaryotes, both or neither? Both. Living cells, by the way, have to have some kind of nucleic acid like DNA. All right. They have to have, well, actually, they have to have DNA. If they don't have DNA, they're not a cell. All cells have to have DNA, but prokaryotes have how many? Okay. How many chromosomes? One chromosome. Yes. All right. What's the shape? Circular. Yes. Prokaryotes have a single circular chromosome. Is it in a nucleus? No. Is it in a nucleoid area? Yes, it is in a nucleoid area. Very good. Eukaryotes have circular or linear DNA? Linear, and it's in a true nucleus, of course. The organism that you are writing your report on is considered prokaryotic, eukaryotic, both or neither. Well, they can't be both. They have to be something. They have to have some kind of DNA. They either have to have a nucleus or nucleoid area. So, since you're doing your report on bacteria, it has to be prokaryotic. Okay, so A. All right. Identify and describe prokaryotic cell structures. Let's go into some details now. All right. Let's start with the shapes and arrangements. Some of you, while doing your reports, have come upon some terms. Maybe they have been a little confusing. So let's go over some of these terms, okay? So one of the things you did need to know for your that you needed to include in your research project is the morphological characteristics. Okay. So, morphological characteristics would include shapes and arrangements. Let's begin with the shapes. So, some basic bacterial shapes include the following bacilli, that's plural, bacillus is singular. Sometimes we call these rods, rod shaped. Okay. Now, some bacteria, by the way, look like this and they are named after this shape. What's a good example of this? Well, how about Bacillus anthracis? Yes, if you've looked at this word and you're thinking, is this the one that causes anthrax? Yes, Bacillus anthracis causes anthrax, and it looks like a bacillus. So the first name, the genus, is Bacillus. Makes sense. All right, now, if the bacterial cell looks very spherical, these are called cocci, that's plural, coccus is singular. So they are spherical shaped, round. Okay, very round. Some bacteria are named after their shape, and if you're probably thinking Staphylococcus streptococcus, you are right. So, two species of organisms here that are coccus shaped. Streptococcus pyogenes, group A strep, or gas, and Staphylococcus aureus. Both have this shape. And so the shape is in the name. All right. Another shape is kind of a corkscrew shape. And these are the spirilla or spirillum, singular, and they are spiral. Now they can be, there's different kinds of spiral shapes. Some are rigid spiral shapes, and some are very flexible. I like to talk about the flexible kind because these are considered spirochetes. They are coiled, they are flexible, and these guys really know how to cause infectious diseases in animals. So this is the one that's the spirochete, okay? Now there are some other shapes out there that are a little bit between the bacillus and the coccus shape. So these are called coccobacillus shaped, okay? Little bit between the two. Really, they lean more towards the bacillus, but we'll go with that. So a very short bacillus shape. Now, occasionally you will run across a shape that is really bizarre. It's bacterial, but holy smokes, what do you make of this? It's branched. And down here it looks very spherical, but here it looks bacillus shaped. And now, if my poor microbiology students on campus saw this, they might assume that they had a contaminated culture. But no, some bacterial types are pleomorphic. Pleo meaning many, morph, okay, morphology is the the morphology is the shape, the structure, okay? So many structured. Okay, it's a many structured type of cell, okay? Many shapes. All right. Okay, now what about arrangements? Okay, so prokaryotic bacterial cells come in many arrangements. Now, some of the most basic arrangements are as following. Sometimes they come as a single. So here we have a single coccus. Right? Sometimes they do appear that way. Sometimes there are two of them and they stay together. The reason they hold together is because of how they divide. They divide and then they just remain stuck together. This is called diplo. This is a diplococci shape or diplococcus singular. Neisseria meningitidis, Neisseria gonorrhea are always diplococci. Strepto. Here we go. So the coccus shape here is in a strepto arrangement. Yes, indeed. So almost like a chain, all right? Typically the chain is fairly long. It just depends. And yes, if you're thinking, oh, there's some organisms named after this, yes. And also, cocci can appear in clusters. Now, this looks like it's getting ready for a pool, a game of like it's getting ready to be on a pool table. These are staphylococci. Trust me, they would rarely come out looking this perfect and this having such geometry. Normally, they look they look a little bit more like a bunch of grapes that you would grab from the grocery counter. Okay, so they're clusters. And yes, some bacteria are named after their arrangements. You probably are guessing some of these. So, for example, Streptococcus pyogenes. Okay, group A strep. They look like this. So here's the strepto arrangement, and they are coccus shaped. So there we are. Staphylococcus aureus. They are staphylo arranged, but they are coccus shaped. So they come out like that. Now, sometimes, unfortunately, the name is deceiving. You might come across something like this, Streptococcus pneumonia. And you would be right to think that it is coccus shaped. You're okay there. But interestingly enough, Streptococcus pneumonia usually appears as a diplococcus. Yeah, I know. Life can be very unfair. It's not my fault. I didn't come up with the name. Sometimes we just continue to do things because we've always done it that way. And nobody wants to step up and say, let's change that. That's really kind of dumb. All right. So again, now let's go to the bacilli. Do they have similar arrangements? Yes, they do. Bacilli can be in a single arrangement. They can be diplo, but it's diplobacilli. They can be streptobacilli, but you will not see staphylobacilli. No, sorry. The closest you can come to this is getting a palisade arrangement. Palisades look like a picket fence. They're side by side by side. This is very unique to some bacteria. In fact, when we talk about the disease-causing pathogenic bacteria, all right, the one that causes diphtheria does this. And it's very unique. So this organism is known as Corynebacterium diphtheria. You don't need to know that for a test, but yes, this is one of those things. If we see this, and if the patient has the symptoms, we do more tests and look for this organism. All right. Now, some of these bacterial structures will mount immune responses, and some are targets for antimicrobials. So let's talk about these. All right. So, when there is a structure on bacteria that is identified by our immune system, our immune cells, we call those structures antigens. All right. An antigen is anything that generates an immune response. And in the old days, we thought that it only generated antibodies in the bloodstream, which is part of our immune response. But later on, we realized that antigens generate a whole lot more. Think of antigens like identification tags. Now, the bacteria and other pathogens would rather not be identified. So they're only ID tags to us or to our immune cells. To them, they would really rather hide these things. They don't want to be identified by the immune response. In fact, they try really hard not to be identified. Okay, now let's look at some of the structures. Sorry, I have neighbors who like loud motorcycles. let's let's look at some of the structures. We'll start on the outside and we'll go inward. The first structure that hangs out here is the flagella, or flagellum is singular. And since it's sticking out there, let me tell you, they are pretty antigenic. There's little proteins that stick off of these guys that will make really good ID tags for our immune systems. So again, flagellum is singular, flagella is plural. What do? They allow motility. You knew that. Of course you did. Now, bacteria have all kinds of different flagella and flagellar types. Okay, and if you look on the side, you can see that. Now, flagella allows a cell to have motility. What is motility? That means they can move under their own power and they move towards good things. And usually they run towards good things, they swim towards good things, and then it also allows them to swim away or tumble away from bad things. Now, how do they know where to go? How do they know there's something good over here and they want to go over there? They have chemical receptors just like you do. Okay, if you're smelling hot cookies in the oven, you have chemical receptors and you might get up and go run and go get some cookies. All right. They have little cellular chemical receptors and they'll move towards the good things, but they'll move away from bad things. Let's distinguish for a moment the difference between motility with a T and mobility with a B. Mobility means that they're moving but not under their own power. Good pathogens are all mobile. Some of them just travel through the bloodstream, but it doesn't mean they're moving under their own power. All right. A lot of bacterial toxins are very mobile through the bloodstream and lymphatic system and through tissue, but they are not motile. They do not have a specific direction. They just go with the flow. So mobility occurs when your patients are pushed around in a wheelchair. Now you know the difference? Okay, good. All right. Now, some types of bacteria have very unusual flagella, very unique, very fascinating. The ones that I speak of are called paraplasmic flagella. These are a little bit, well, if flagella were like hairs, these would be ingrown hairs. These little types of flagella wrap around the body of a very elongated bacterial cell. You see these paraplasmic flagella only in spirochetes. Remember the flexible spiral kinds of bacteria? Oh, yeah. So these flagella are like filaments and they wrap around the cell and they're sort of underneath a bit of a sheath. Now, the types of bacteria that are spirochetes that have paraplasmic flagella include the following: Treponema pallidum that causes syphilis. Also, Borrelia burgdorferi that causes Lyme disease. Little side note about Borrelia. which you see over here. It moves again through the skin, through tissue, down through basement membranes, into blood vessels, and all around. They get in and invade most all tissue, including the brain, and they can do this within 48 hours after entering the host. Scared of Lyme disease? Should be. Do you think motility is a virulence factor for these guys? Oh, yeah. Let's watch the video. Now, here you can see them spiraling away. Imagine them drilling through tissue and getting, well, throughout the organ system, including the nervous system and even the central nervous system. Now, here's a close-up view, and you can see how the paraplasmic flagella are wrapped inside, and you can see how it just spirals. It's actually one flagellum on one side, one on the other, and they wrap, and they meet in the middle, and they move a little bit like actin myosin filaments. Okay, very interesting. I love that video. So again, yes, it's going to flagella are really going to increase a bacterial's ability to cause disease. So, and that's what a virulence factor really is. A virulence factor is something that increases the organism's ability to cause disease. So this is the first virulence factor we've talked about. Okay. All right. Now let's talk about another structure. Again, moving from the outside, moving in. These are pili. Pilus is singular, pili is plural. Some bacteria have these, okay? And what do? Well, there's a few different kinds. And there's sometimes we call these structures fimbriae. I just call them pili just to cut through just a lot of information that you don't know or don't need to know. But some pili are sex pili. They are hollow tubes and they allow DNA to pass from one bacterial cell to another during sex. Okay, so if one bacterial cell loves another bacterial cell very, very much, what they might do is pass some of their DNA in the form of a plasmid through the sex pilus into the next recipient bacterial cell. So this is called a sex pilus. All right. So here's another illustration, and you can see the plasmids that are made. They can be they can multiply and pass from one bacterial cell to another. So this is one way from one bacterial cell to share genetic information with another bacterial cell. If you remember earlier, I said that plasmids can contain information on how to make toxins, how to become resistant to antibiotics. So up here, maybe this bacterial cell didn't know how to be antibiotic resistant, or it didn't know how to make a toxin. Now all of a sudden, through bacterial sex, it does. Now, pili also allow bacteria to attach to substances. Gram-negative bacteria, which we will talk about soon, are very good at making pili. And yes, they will allow these bacteria to attach to and hold on to a lot of different structures, including fresh vegetables and fruit. So when E. coli is in water and the water is used to wash vegetables or to water the plants when they're in the ground. All right, well, E. coli has pili and it can very easily attach to the vegetables. And believe me, they will hold on very well. This is why it's hard to wash out E. coli from vegetables. This is also why so many problems occur, and there are E. coli outbreaks from restaurants, especially fast food restaurants. Sprouts, lettuce. There was a romaine lettuce problem that occurred this last summer, because yeah, E. coli was attached to it and wouldn't come off. And normal washing isn't going to do it. Sorry, you can sit there and rinse all day under the faucet. Won't work. Okay. So, and it also keeps E. coli from being washed out of your urinary tract when you urinate. So, do you think that this is a virulence factor? Oh, yeah. Here's a very good example of what I'm talking about here. Now, it turns out E. coli is the number one cause of urinary tract infections. Oh, yes, it is. So, let's say that it gets up the urethra and here we have the urethral, the epithelial cells, and using the sex pilus in this black bar, yes, I made this, it swims over and attaches to the lining of your urethra or to the bladder. Okay, now you can urinate, mostly it's the urethra, but you can urinate and urinate and urinate, and they will hold on. Bummer, huh? So then you need an antibiotic. Now, I'm going to talk about cranberries. If you're thinking, okay, what? Well, if you know, we tend to give patients with urinary tract infections, UTIs, cranberry juice. It has to have real cranberries in there. Why, do you ask? Well, now I'm going to reveal the reason to you, right to you. Cranberries, like a lot, well, actually, there are a lot of fruits and a few vegetables that have proanthocyanidins. I know you're like, what? Gesundheit? Yeah, it's a chemical that is, cranberries are high in this chemical. And they tend to attach to the pili and block the pili. So if our E. coli has this, cranberry proanthocyanidin attached to its pili, is it going to be able to attach to you? Not so much. Oh, sorry, not so much. I thought I had one more animation going. I do the best I can. Yeah, so it can't attach to your epithelial cells very well. So we tell patients to drink a lot of cranberry juice. Not only will it help flush out stuff as best you can, but also it will block E. coli from attaching. So this is something that we tell patients to do. Now, it doesn't work on all E. coli. E. coli have different pili. Yeah, sorry. But it does work on a lot of them. So how is it? Now, if you remember your anatomy, you remember that, okay, um, that the urinary tract is different from the bowels and the anus. And E. coli is a microorganism that lives in the colon, and that's why it's called E. coli. Now, why is it that an organism that lives in the colon is the number one cause of urinary tract infections? Well, it's quite the swimmer. Okay, it has flagella, what we call paratrichus. Okay, lots of flagella. It's a heck of a swimmer. But still, how is it getting from the anus up the urethra? And why, doggone it, are women more prone to get urinary tract infections than men? Well, that's a good question. First of all, let's just talk about the mechanism. All right, so urinary tract infections. Females are more prone, they're more at risk of getting urinary tract infections because they have a small urethra and because the anus is very close to the urethra. Hmm? Whereas boys have a longer urethra and it's a longer distance to swim. You have to be careful of bath water too, because this is okay, the bath water and the baby's here, and E. coli is a heck of a swimmer. Once it gets into the urethra, it can into the epithelial cells of the urinary tract, it will hold on. And it turns out also that young girls, they don't have a lot of normal microbiota yet. Turns out our normal microbiota will fight off some of the E. coli, stop them from attaching. um, Older women will get these UTIs later on, maybe because they've been taking antibiotics and they've lost their normal microbiota. Um, it turns out, again, uncircumcised boys are also very prone to getting E. coli UTIs. It's more hygienic to have the circumcision. Um, and for reasons that I cannot even begin to explain, Caucasians are more susceptible to urinary tract infections. Sorry, couldn't tell you why. Oh, there needs to be a bracket there, huh? Okay, so I'm there. Let's talk about the next structure down. Some bacteria, not all, have something called a glycocalyx. What is that? Well, whenever you see glyc, maybe you think, is that sugar? Is that a sugar like glucose? Yes. Yes. Think sugar when you see GLYC. Think sugar. Calyx is like a coat. Whenever you see that term, there's botanical terms also that talk about certain flowers that have a calyx. But yeah, sugar coat. That's what glycocalyx is, sugar coat. Now, there are two kinds that are relevant to microbiology and bacteria, okay? The first kind of glycocalyx is known as a slime layer, and as the name suggests, it's slimy. So this is a sugar coat that is loosely attached to the cell and is very, very slimy. And here you can see this bacteria in this slime layer. And they manufacture this and send this out. Okay, so why do they make this? And by the way, if you want to know what a slime layer is, what it's like, just run your tongue over your teeth. And if you haven't brushed your teeth, in the last few hours, you're probably going to feel the very first, the beginnings of a slime layer being made, and then that can develop into something called a biofilm. So the glycocalyx is there to prevent dehydration. Bacteria don't want to dry out either. And it does add a layer of protection. So again, and it turns out that these bacteria that start to form a biofilm, um, they will talk to one another and they will say, "Hey, is there enough here? Should we form a biofilm?" Dentistry is all about preventing biofilm, by the way, getting rid of these kinds of glycocalyx structures and just getting rid of all of that. Now, just a little side note, bacteria hiding in a biofilm are about 1,000 times harder to kill. If you're thinking that this only happens in your mouth, think again. Bacteria that live in your body that invade are always forming biofilms. They do this in your lungs, they do this in your colon, they do this, if you have a pacemaker put in or a catheter put in, they will start making biofilms. And again, they are 1,000 times harder to kill. Do you think that's a virulence factor? Oh, yes, it is. So this is why dentists tell you to reduce, reduce, reduce. Don't let the bacteria remain there. They're going to be forming slime layers. They're going to start making biofilm. So you brush, you floss, and you see your dentist regularly. Okay, this is a scanning electron microscope picture, and you can see the covering of the bacteria. And by the way, when they gang up on you, they don't even have to be the same species. They do talk to one another. They send out chemical signals like hormones. This is called something called quorum sensing, by the way. All right. The second type of glycocalyx is known as the capsule. What is the difference? The capsule is very tightly attached to the cell. It's like a really tightly attached layer. And this really gives bacteria, especially the pathogens, the ability to cause disease. So, this is very much a virulence factor. Why? As this is tightly attached, not only is it hard for drugs to enter, but also the capsule hides the ID tags of the bacteria from the white blood cell soldiers. So if the white blood cells are coming around looking for antigens, ID tags, they won't be able to see them or won't see them very well. And bacteria that have these are they're hard to respond to immunologically, meaning your immune system does not respond to this. Also, it's hard to get even a vaccine to work well when the bacteria have a capsule. For example, all right, in the back of your nasopharynx, you might have a little bacterial organism known as Haemophilus influenza. Now, Haemophilus influenza comes in two varieties. The Haemophilus influenza that doesn't make a capsule is not invasive, tends to cause mild disease, maybe maybe a little bit of epiglottitis, but it's not really a big deal. The one that does make a capsule is what we call Haemophilus influenza B or Hib, and we have vaccines against that one. This is the one that becomes invasive and can cause some very serious diseases. So if Haemophilus influenza doesn't have a capsule, not as virulent. If it has a capsule, very virulent. It invades tissue. One of the things that Haemophilus influenza can do, and by the way, this is Haemophilus influenza. Okay, sorry. Or no, I didn't. Yeah, sorry. It causes something called meningitis. So it can get into the meningeal lining. That's the lining around the brain, right? It causes an inflammation, and we call this meningitis. All right, meningitis. So there's a lot of different organisms that cause meningitis, not just Hib, but I'm just saying when this bacterial organism has the gene that allows it to make a capsule, it is invasive and it is potentially dangerous and deadly. Okay. Now, let's go to the next structure. This one is called the endospore. I had to draw my own little green structure in here. Not all bacteria make endospores, but some do. And endospores are very, very important and they're medically significant. Here is an actual picture of a bacterial cell, and here at the very end is a little endospore. It's like, what's that little hole there? It's like it didn't get stained or something. That is true. This is not a nucleus. This is an endospore. Remember, bacteria do not have a nucleus. Endospores are extremely tough, very resistant. This stage of bacteria is the toughest living form that we know of on this planet. When it comes to living things, if it's in an endospore form, it is the hardest thing to kill. They are essentially immortal. Okay? We know that some of them live millions of years. Okay. Nothing is considered a sterilizer unless it's able to kill endospores. When we have autoclaves set up to sterilize surgical instruments, we test it. And if it can't kill an endospore, it's not working. Okay. Chemicals that we use, very few chemicals are able to kill endospores. And again, if they cannot kill an endospore, they are not sterilizers. All right. We kind of set the bar at endospores. Okay. Does it kill an endospore? Yes. Then it sterilizes. All right. Bacteria. What is this exactly? So this is a stage, a form that bacteria go into when there is a threat. Typically, it's a lack of food. Okay. It's a response to a threat. And oh, let me move back up here. So this little image here, this bacterial cell is forming this endospore because it is feeling threatened. In the laboratory, if we want to see endospores, and by laboratory, I mean my laboratory at Clarkson College, if I want to see an endospore in the bacteria, I have to make the bacteria miserable. All right, we need to just stop giving them food and they will start to go into this phase. All right. So what bacterial types make endospores? Now, there are lots of bacterial types that do make endospores, but there are two basic genera, those are the genuses, that are medically significant that make endospores that you need to know. So if they have the first name Bacillus, that's the genus, Bacillus, or the first name Clostridium, okay, the genus Clostridium, they are endospore formers. So I'll throw out some examples here. Bacillus anthracis causes anthrax. We already talked about that one, right? Because its first name is Bacillus, it means it makes endospores, and it does. Clostridium. Are there any Clostridiums that we know of? Because they do make endospores, so they're medically important. Clostridium tetani causes tetanus. Clostridium botulinum causes botulism. Clostridium perfringens, you probably don't know that one very well, but it causes gas gangrene, one of a few. Clostridium difficile, C. diff. Okay, all of those make endospores, which are essentially immortal, and they're very, very hard to kill, and they don't die by ordinary means. All right? All right, so let's go into how bacteria go into these phases. Typically, bacteria are just being bacteria if the environment is good. So there's plenty of food, it's very safe, and they're happy. So we would say they're in the normal bacterial stage or the vegetative phase of their life. And here they don't have any endospores. They're just eating, they're metabolizing, they're dividing, so they're undergoing reproduction, which we'll talk about a little bit later on. But let's say we made them miserable, or they ran out of food, or there's some kind of threat, maybe a high temperature, lack of oxygen, or presence of oxygen in some cases. All right, then what it will do is it'll form a very tough spore coat around its most vital components. Now, what would that be for a cell? Well, cells must have DNA. All right, so that they would make a copy of DNA and put that into an area where the spore is going to form. Ribosomes, because all cells have to have ribosomes like they do DNA to make proteins, so those get thrown in there. If there are essential enzymes, those get thrown in there. So let's look down here, and this would be number 3. And here you can see where the little endospore is being formed. And now the cell will break down. What's left of the cell, it breaks down, and the endospore is free in the environment. And it has a very tough coat that has wrapped around the essential components that I talked about. Okay. So the cell has broken down, and this would be here we'd have number 2, and the endospore is released. Okay, it's out in the environment. So here we can see the normal vegetative cell. Here's the cell feeling threatened, making an endospore, and here's an endospore released into the environment. And remember, so this is not undergoing metabolism, it's not eating. It's not moving, it's not doing anything. It just survives. And it blows around in the wind and it gets on everything. And this is like a hibernation of a bacterial organism. Okay, now let's say the endospore lands in a really happy place again. Well, it will actually sense it. Usually it's food, and it will return to its vegetative state. It goes back to number 1 down here. Okay, so again, what bacterial types make endospores? What do you need to know? Well, like I said, there are bacteria that belong to the gram-positive groups. Talk about that in a little bit. Again, the first name is Bacillus or Clostridium. Typically, we'll say this, by the way, whenever you see this while you're reading journal articles, this means some species of bacillus. So this is bacillus and a specific epithet. So this could be Bacillus anthracis, but it could also be, oh gosh, several others. We won't go into it, but Bacillus anthracis is the only one you need to know that causes anthrax. So that's pretty scary, right? Yeah, because we know how anthrax is. We know it's a bioweapon, don't we? Clostridium. There are many species of Clostridium, including Clostridium tetani, causes tetanus or lockjaw. Clostridium botulinum causes botulism. Clostridium difficile, known as C. diff, causes C. diff membranous colitis. And if you've been in a clinical setting in any way, you know what this one is. All right. Remember the anthrax powder, right, after 9/11? And everybody was worried that anthrax was still being sent through the mail. And some of it was being sent to the New York Post. Tom Brokaw, who was was a journalist at NBC, a couple of senators, and yeah. So these are endospores. What is the toughest thing to kill? Endospores. Yeah. Little side note here. Now, endospores we know can survive in alcohol for over 100 years. Yeah, and by the way, 70% is the most effective kind of alcohol, but it still isn't going to work. So, yep, you could take it out of the alcohol and grow it up on a petri dish. It'd be fine. So, let's look at alcohol-based hand sanitizers for a moment, shall we? What is their active ingredient? Okay, so they have inactive ingredients, which is pretty much water and gooey stuff. So this is the only thing that they're using to kill your bacteria, and it is ethyl alcohol, ethanol. It's alcohol. Okay, 70%. Is that going to work at all? No, not at all. Will this work? Well, nothing is going to sterilize the endospores. And let me tell you, when it comes to C. diff, it will be everywhere. The only thing you cannot sterilize your hands. It isn't going to work. And we're not going to put any chemical on your hands to sterilize your hands. It's too caustic. But here's what you can do. Soap and water, okay? Soap will help break up any kind of loose cells, any sticky stuff that might hold on to the endospores, and you wash and you know, sing the ABCs in your head, rinse really well, and rinse the endospores down the sink. You're never going to sterilize your hands, but you can reduce, and give your next patient a good old chance, okay? Instead of carrying it from one patient and their room to the next. Okay. Endospores, a virulence factor? Yeah, surviving indefinitely is a pretty good virulence factor. All right. So again, if your patient has C. diff, and this is Clostridium difficile in here, the endospores being formed. Yeah, that's bad. All right. Well, now that you're freaked out, moving on. Still moving from the outside, from flagella all the way in, the next layer is the plasma or cell membrane. All cells have to have a cell or plasma membrane. If the cell membrane loses its permeability, that is the end of a cell. Your cells have them, my cells have them, your cat, your plant, your dog, everybody has them. All the bacteria, all the pathogens, the living pathogens, they all have membranes. Okay? Now, let's talk about this really quickly. Hopefully, you do remember that plasma or cell membranes are semi-permeable barriers, and they are made up of a phospholipid bilayer. Amongst the phospholipids, you will have different kinds of proteins in there with various functions. Some of the proteins have sugars sticking out of them. By the way, they make pretty good ID tags. Both the proteins and the sugars do. But I want to stress that a cell membrane is not a cell wall. Do not mix these up. It becomes really important a little bit later on. A cell has to have a plasma or cell membrane. A cell does not have to have a cell wall. Our cells don't have a cell wall. We don't need them. We've managed without them. But yes, cell walls are different than cell membranes. Right now we're talking about cell membranes. Okay. Now, again, cell membranes are like all cell membranes in that they have simple and active diffusion that regulates the movement, of course, of nutrients and waste. And of course, gases diffuse in and out without any trouble. If you have lipid soluble materials like vitamins, they move in very easily. Anything that's big and not water soluble is not going to move in. All those things that Mr. Seberg has taught you. Now, again, keep in mind, all cells have to have a membrane or they're not going to survive. So, yeah, if anything happens to disrupt a membrane, the cell dies. There are drugs that disrupt cell membranes of bacteria and fungi. But the problem is that, okay, they're more toxic to bacteria and fungi, stressing the word more toxic. They tend to have a lot of toxicity problems, okay, because the enemy have membranes, so do we. Finding one thing that is really, really, really unique to poison and to destroy is very hard, and in some cases not possible. Okay. So let's look at some other morphological structures and processes that are targeted by antimicrobials. Why are you learning so much about cells? And a lot of students kind of tune out in when we talk about cell morphology. Maybe you were jotting down notes because a lot of those morphological characteristics are relevant to your pathogen and your pathogen topic. Um, but again, why go over this really? Why do you need to know cell structures? Because if you are going to kill a pathogen, you are going to have to understand how it ticks. You're going to have to target a very unique structure in that bacterial or fungal cell or protozoan, whatever it may be. So let's say the bacteria or the fungi or whatever are inside your patient. You're going to have to go and give them a poison, aren't you? You're going to have to give your patient a poison. Hopefully, it's not a poison that attacks them, it's a poison that attacks the bacterial cell or the fungal cell, but doesn't attack their cells. Okay? And a lot of the processes and structures that you've heard about are great targets. We can target structures and processes that are unique to these pathogens or relatively unique. That's why we go through this. All right, ready? Do you need to go get some more caffeine? Okay, go get some more caffeine. Come on back. All right, we're back. All right, so let's talk about the cell wall. Cell walls are unique to some pathogens. Now, do you have a cell wall? No, you do not. So maybe we could go after some process that makes the cell wall of like bacteria. Yeah, if we could do that, if we can target a structure or process that makes bacterial cell walls, okay, that will leave us intact. Our cells will be fine, but the bacterial cells will be destroyed. Now, most bacteria have a cell wall, and its major component is peptidoglycan. Remember that term, peptidoglycan? Well, we're back. All right, so. What is the cell wall? Remember, it is not a cell membrane. It isn't semi-permeable. No, no, no. It's a very strong, rigid structure, and it prevents the cell from exploding. Here's a little, I'm going to go to, ah, oh, shucky darn. I do have another animation that I can show you. I was hoping this would get to the website. All right. So, in bacteria, the cell wall is made up of peptidoglycan. If you break this down, peptide, like peptide bonds, oh, it must be part proteinish. Yeah. Glycan, remember GLY must be part sugar. Yeah, peptidoglycan, that's what it's made of. Peptidoglycan is only found in bacteria. So, yes, it's very unique. This makes it a great target, right? The peptidoglycan structure distinguishes actually two types of bacteria, and these are the ones we've been talking about. And I said I'd talk about them earlier or later. Okay, it's later. So, gram positives and gram negative bacteria are in those categories because of the peptidoglycan cell wall. All right, so gram positives and gram negatives. They belong to one or the other groups according to the type of peptidoglycan cell wall and and structure that they have, okay, the cell wall structure. So, what is this gram positive, gram negative stuff? Please explain. Glad you asked. It's all about the bacterial cell wall. And again, why is this important? Because it's unique. This is going to tell us a lot about our bacterial organism, which is going to tell us a lot about how to treat the bacterial organism. All right, to talk about gram positive and gram negative bacteria, we have to talk about this guy. This is Hans Christian Gram. He developed a staining system. Actually, it's a chemical type process that just happen to have, it has colors to it. So in a laboratory, you're going to have different colors come out depending on what kind of bacterial cell you're looking at. All right. So, Hans Christian Graham developed the Graham stain. Hans Christian Graham realized that certain chemical stains react with different bacteria differently, and this allows us to differentiate between two types of bacteria. Okay, so we're going to be able to understand cell wall structures. What's the procedure? Really quickly. Okay, so number one, it wasn't supposed to come up together, but all right. So first of all, you add a chemical that happens to be purple. And this is the very first stain. This is the one of the critical stains. This is called crystal violet. It's a primary stain. And when it hits a slide of mixed bacteria, all right, everything's going to look purple. Well, because it's a purple stain. Even if you rinse it off, the crystal violet will sort of hold on to everything and still look kind of purple. It is a stain. But the next step is very unusual. The next step is we add iodine to the crystal violet. Now, this does something strange. Now, violet is like a purple color. What's with the crystal part? Well, once you add the iodine, the crystal violet crystals form. The crystals, when they form, they lodge themselves in the peptidoglycan cell wall. All right. Now, if there's a lot of cell wall material, the crystals get in there and boy, they're going to get stuck. Even if you add the next step chemical, which is the decolorizer, it's an alcohol and or sometimes it's got acetone in it. But when you add the decolorizer, if there's not a lot of crystal violet attached to a lot of cell wall, well, the bacterial cells will just turn clear. Now, there's the last step, which is the counter, and we'd rinse very well. Okay, the last stain is called the counter stain, and actually Hans Christian Graham didn't come up with this one. Another scientist did, who said, I'd kind of like to see what the other cells might look like. The ones that rinsed out, that didn't have a lot of cell wall and didn't maintain all that crystal violet. So that scientist said, let's add a different color for contrast. So he added a red saffron in. And okay, so all the cells that were washed out before, decolorized, now look red. Okay, so the ones that held on to the crystal violet are considered gram positives. The ones that did not are considered gram negatives. Okay, come on, wake up. There's significance, I promise. So this is what it would really look like under a microscope. You have your gram positives, like, oh, Staphylococcus, Streptococcus, Bacillus, Clostridium. They all are gram-positive bacteria. And then you have gram negatives that would include, oh, lots of things like Borrelia burgdorferi, Treponema pallidum, E. coli, Salmonella, Shigella, a lot of Haemophilus influenza, lots of them. All right. So right now, this really didn't tell you much except for some turned purple and some became red. Okay, what is this telling us, though, about the morphology of the bacterial cell? Because that's really what we're trying to do here. Okay, so gram positives, if it's a gram positive bacterial cell like this one, okay, let's say this is Staphylococcus aureus or Bacillus anthracis, and it's got lots and lots of layers of peptidoglycan. The crystal violet crystallizes in here and gets lodged and it gets stuck. So gram positives have a lot of outer peptidoglycan in the cell wall. That's where the crystals form and it doesn't wash out. Cells remain purple even when you add the red saffron in. Okay, and they look like that. And that's Staphylococcus aureus, by the way. All right, gram negatives. Now, this is something that is so unique. Gram-negative bacteria do not stain purple because, okay, they don't have a lot of peptidoglycan. Look, it's just a couple of layers. But not only that, they have a very freakish outer membrane. Now, wait a minute. Now I've lost you. I know. Back up. Do you remember all cells have an inner plasma membrane, a cell membrane? Yes, all cells have that. This one does, and this one does. Gram negatives have a second outer membrane. Have you ever heard of a cell that does this? Ever? No, because no other cells that we know of on this planet have a second outer membrane. Okay, just these bacteria that we call gram negatives. It is very protective. Any chemicals like stains have to get through an extra layer, protective membrane layer. All right, so things don't penetrate through this layer very well. Okay, so gram negatives, they have an outer protective membrane, not a lot of peptidoglycan. The crystals and crystal violet do wash out that gets in there. And then the counter stain makes them look red. All right. So these are E. coli, by the way. Okay. So, term here. When we talk about the cell envelope, we're talking about anything that surrounds the cell. So for a gram-positive bacteria, it's one cell membrane and the cell wall. Okay. If we're talking about gram-negatives, you have an inner membrane, an outer membrane, and sandwiched in between is the peptidoglycan. Okay, of the gram-negatives. All right. So, little bit more. Let's look at the structure. So it seems to all be about the peptidoglycan, right? The cell wall. And I said gram positives have a lot of these layers. Do gram negatives have peptidoglycan? Yes, just not as much. Okay, but that's okay because they have a second outer membrane. Let's look at the structure. So peptidoglycan, again, there's peptide chains and there are sugar bars. So the peptide chains are there in sort of an orange color. The sugar bars are in pink and blue color. Here, if you can't, if the colors don't come out very well, I'm just going to point to the sugar bars, okay? And so you have links between the sugar bars here are actually, it's a disaccharide sugar. And it's made up of N-acetylmuramic acid and N-acetylglucosamine. Yeah, I know. You only have to know NAG and NAM, all right? So NAG and NAM and NAG and NAM, that makes the sugar bars. And then they have these cross bridges that are the peptide chains. These are important. I know you're thinking there's no way this is going to be important. Yeah, there is a reason for this. All right, so this is a little bit easier to see. It doesn't show everything here, but the gram-positive cell walls have a thick outer peptidoglycan layer sticking out. And again, you have the sugar bars, NAG and NAM, and then the cross links here of peptides, okay? All right, and that's your gram-positives. Now, gram-positives, many layers of peptidoglycan. There is one other thing, though, that I wanted to mention. In addition to the just single membrane and this thick peptidoglycan cell wall, gram positives have this chemical called teichoic acid, and it sticks up from the cell, well, through the cell wall. And this is like a gram positive identification tag, or we would say it's an antigen. Teichoic acid is an antigen. I know it's not showing here, but just know it sticks up and it identifies gram-positive bacteria to our immune system. So again, it attaches the cell wall and the plasma membrane and it sticks up and it contains antigenic specificity. It generates an antibody and immune response. Now, gram positives, these guys are very susceptible to penicillins, and there are many types of penicillins. But the first penicillins, yes, they were really good at targeting our gram positives. And what do they really do? The penicillins are going to attack an enzyme that links the peptidoglycan together. Okay, so penicillin links the enzymes that link peptidoglycan. My example is Staphylococcus aureus. Okay, know that this is a gram-positive example. It's like the poster child. Gram negatives. Again, it has a cell wall, but it's thin, just a little bit of peptidoglycan. But it has an outer and an inner membrane. The outer membrane is freakish, unique. I use freakish just to put it into your head. It's very protective, isn't it? It's got not only one semi-permeable membrane, these bacteria have two semi-permeable membranes. Again, do any other cells have two membranes? No. None. Zilch. Nada. Only gram-negative bacteria, like E. coli. Okay. Now, there's a little more to that. You have, again, your inner, your outer membranes. The space in between is called the periplasmic space. Some textbooks will say gram-positives have a periplasmic space. No, wrong. It has to be the space between the two membranes, and only gram-negatives have that, with a thin layer of peptidoglycan. That outer membrane is very protective. Many gram-negatives are not hurt by penicillins. Okay, penicillins won't work on a lot of gram-negatives, actually the majority. So we tend not to give patients with a gram-negative infection a penicillin. Okay? Now, in addition to being protective and freakish, this outer membrane has a chemical substance known as lipopolysaccharide, or LPS. Only gram-negatives have LPS. This is medically significant. It is an ID tag. It's an antigen like teichoic acid, but teichoic acid is gram-positives. This is gram-negative. LPS is an endotoxin. That is a toxin that is embedded in this outer freakish membrane. Does any other bacterial organism have an endotoxin? No, only gram-negatives. Does a gram-positive have it? No, only gram-negatives. Only gram-negatives have LPS in this outer freakish membrane. Endotoxin. So what does this do exactly? It doesn't sound like it would be a lot of fun in the patient. Well, in small amounts, it works as an alarm system to your immune response, and in small amounts, it causes a fever. Again, in small concentrations. In large concentrations, it'll cause your patient to go into shock and possibly kill them. So, yeah, that's not good. More about this in a little bit. Okay. Okay, so which of the following types of bacteria is lacking a cell wall? Gram-positive, gram-negative, none of the above. You said none of the above, right? Remember, gram-negatives, it's true, gram-positives have a lot of peptidoglycan, but gram-negatives have, the gram-negatives have a little bit of peptidoglycan, but it's there. They do have a cell wall. Okay. So, oh, that wasn't supposed to be there yet. I'm going to skip this one and come back to this. Let me just back up for just a moment. And I'm going to go back to one of the original slides because right now your head is hurting, right? I'm going to help a little bit. All right. All right. Here's our gram positives. Here's our gram negatives. Now, remember this. Repeat after me. Gram positives stain purple because they have a lot of outer peptidoglycan and they're sensitive to penicillins. Yeah. Oh, yeah. Gram positives, come on, say it after me. Gram positives stain purple because they have a lot of outer peptidoglycan. They're more sensitive to penicillins. All right. Now, once you get that in your head, again, do not be confused. Gram negatives have peptidoglycan, but just not a lot. Now, gram negatives, it's a little harder to remember them, unfortunately. And unfortunately, they are they're even trickier. They are harder to kill. They have an extra outer protective membrane. This makes them harder to kill. Okay? Protective outer membrane. Gram negatives have the LPS, which is an endotoxin. Right? There are other ways, like I said, you should know if the gram negatives have the LPS, the gram positives have teichoic acid. All right, make sure you know the difference. And again, which one would we treat with penicillin? Gram positives. You'd use something else usually for E. coli. All right. Okay, which one has the most outer peptidoglycan? Gram positives. Which one has a little bit of peptidoglycan? Gram negatives. Which type of cell on this planet has a freakish second outer membrane? Gram negatives. Good. All right. Got it? Make sure you do. I wanted to mention too, we're going to talk about how penicillin works. It actually blocks the enzyme that links peptidoglycan together. That's what penicillin does. All right. Let me just move forward. All right. So that's the end of cells part 1. If you need to go take a break, go take a break. And we're going to begin cells part 2. Yay! Okay, welcome back. This is cells part 2. So, all right, just to recap, which type of bacterial cells have a freakish outer membrane? Gram positives or gram negatives? Gram negatives. All right. So, what's in that outer freakish membrane? Teichoic acid or LPS? LPS. LPS is an endotoxin, right? What happens if I have a little bit of LPS? Fever. What happens if I have a lot of LPS? Shock, possibly death. All right. So what are good examples of gram-positive bacteria? Okay, that would be like Staphylococcus aureus and Streptococcus and Bacillus and Clostridium. All right. Gram negatives would be things like E. coli. All right, Treponema, the spirochetes. Okay. All right. So what color is a gram-positive bacterial cell going to be after a gram stain? Gram-positive stain, purple, right? Purple. So do they have a lot of peptidoglycan, a little peptidoglycan, or no peptidoglycan? Gram-positives have a lot of outer peptidoglycan. Do gram-positive bacteria respond well to penicillins? Yes, they do. Because gram-positive stay purple because they've got all that outer peptidoglycan and they are susceptible to penicillins. All right, so that's the P's, right? That's how you remember what is what. The gram-positives are easier to remember because of this, I know. The gram negatives, you're just gonna have to memorize it. I don't have as much. I don't have anything clever like I do with the gram positives. All right, so now let's talk about a little enzyme that makes peptidoglycan. Why is peptidoglycan so important anyway? This cell wall material made up of peptide chains and glycan sugar bars. I mean, what's the big deal? Remember that if you're going to treat a bacterial cell, you're probably going to have to provide a poison for that bacterial cell, right? We call it an antibiotic. So your patient has a bacterial infection, you're going to give them an antibiotic, and it's going to go systemic. It's going to have to go in and it's going to have to poison cells. The point is not to poison the human cells, poison the bacterial cells. So what do the bacteria have that we can target? Well, to start off with, they have something really super duper unique. They have cell walls. We don't have cell walls. And on top of that, their cell wall is made of peptidoglycan. So man, even if humans had cell walls, it sure wouldn't be made of a peptidoglycan. But what would the drug literally go after and attach to? Well, how about one of the enzymes that makes the peptidoglycan? Aha, and that is a key point. So this enzyme that of which I refer to is called the PBP enzyme, and it is a huge, big antibiotic target, and we're going to talk about it a lot. All right, so the PBP enzyme, here it is. And like all good enzymes, it has an active site and what fits into the active site, but its normal substrate. And its normal substrate are some of the peptide cross, the peptide cross bridge chains. And that's made up of actually 5 amino acids. You do not need to memorize the amino acids, but the last couple fit perfectly into the active site. So that would be the that would be the substrate, right? Yeah. So actually, it's alanine. Alanine, dealanine, dealanine fits in there perfectly. So pretend for a moment this enzyme has a really cool name, and PBP stands for peptidoglycan building protein enzyme. Okay, PBP, because that's kind of what it does, right? And we're going to pretend that's what the PBP stands for because it's easier to remember that way. It is unique to bacteria. So if the bacteria is in your patient and you're going to send a poison after it, go after this. Okay, go after this. So the PBP function, again, it links the cell wall peptide chains, which is the enzyme's normal substrate. Without PBP linking together the peptide chains, the cell wall, well, it's going to be weak. What happens if the cell wall is weak in a normal hypotonic environment? Well, give you one. That's, yeah, I know. It undergoes cell lysis. It'll explode. Water rushes in, and the cell won't be able to handle it, and it'll explode. Okay, let's kind of disclosure here. Let's say what PBP really stands for here. Now, you can think of PBP as peptidoglycan building protein because that's what it does. But that's not what PBP really stands for. When the scientists were trying to figure out what penicillin does, because they knew penicillin killed bacteria, right? And they were trying to find the target of penicillin. And they realized, oh, penicillin was a competitive inhibitor, or basically it attached to this little enzyme here. So, because they were doing research, trying to figure out what penicillin stops, they name this little enzyme the penicillin binding protein. I know. They found the enzyme while working, trying to figure out how penicillin worked. But penicillin interferes with peptidoglycan production. All right. Penicillin is a competitive inhibitor. So so over here, we have these, they look like peas, but they're actually supposed to look like bees for beta-lactam. So these guys, penicillin, jams up the active site of our PBP enzyme, and that's what it does. So penicillin is a competitive inhibitor that outcompetes the peptide chains. And there it is. It outcompetes the peptide chains. And the peptide chains, the normal substrates, then don't fit in the active site. And so therefore, this little PBP enzyme in gray can't do its job. And so the peptidoglycan doesn't get its cross links or its cross bridges linked up. And okay, and again, it's going to be more effective against gram positives. Why? Because they have all that peptidoglycan, but also they don't have a protective outer membrane. Okay? And gram negatives have that freakish outer membrane that not only makes it hard for the penicillins to get in to attack our little PBP enzymes, it also, even if the peptidoglycan doesn't get linked up, that outer freakish membrane sort of keeps the bacterial cell from exploding. So that extra protective membrane is very useful for gram-negative bacteria. Okay, So again, outer membrane of gram-negatives block the access of penicillins and keeps the cell from exploding, which I don't have that line there, but that's okay. Remember I said I would show you another animation. So this is E. coli. Um, well, or let's just say these are bacteria. Long story behind that. So here are some bacteria, some bacilli, and they are replicating by a process called binary fission. One cell becomes two, two become four, and they look like happy little bacteria, don't they? And they have plenty of nutrients. And now try to picture the little PBP enzymes in there trying desperately to link up the new cell walls as the cell is growing, right? So new cell walls have to be manufactured and little PBP enzymes are busy in there. Okay. Now, let's add penicillin that'll block the PBP enzymes. What's going to happen? Pop, 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 pop. Okay. This is osmotic lysis. The water rushes into the cell and the cells explode. So penicillin, imagine it going in and blocking the PBP enzymes, and the cell wall's unstable, and pow. This is why we give our patients penicillin when they have like a staph infection or sometimes a strep infection. This is what we want to see. So this can go in the body. So would this penicillin hurt our cells? No, there's nothing for penicillin to attach to. Okay, penicillin attaches to the PBP enzyme that links together peptidoglycan. There is no target in a human or eukaryotic cell at all. Okay, so pound for pound, there really isn't a better antibiotic than penicillin. It's amazing. Unless, of course, you're like me and you have an immune system that thinks of it as an invader and tries to destroy it, and then you have an allergy. So allergies are different. Totally, totally different. But when it comes to killing a gram-positive bacterium, yes, it's fantastic. Now, are there some gram-negatives that are treated with penicillins? Yeah, gonorrhea originally, before gonorrhea started mutating and being resistant to penicillins, is treated with penicillins. But for the most part, we don't do that. Usually it's gram positives are treated with penicillin. All right? But there are always exceptions. All right. I know you heard that the tuberculosis bacterium is an acid fast bacterium. You heard that somewhere, I'm pretty sure. What is that? So this is a different kind of envelope that we're going to talk about. So it's all about the cell wall and the cell envelope. But okay, the Mycobacterium tuberculosis bacterium has the toughest outer protective layer of all. So one thing let me just back up one moment here. When it comes to gram positives or gram negatives, which one do you think is easier to kill? The well, that's a bad image. So, so yeah, the easier one to kill is the one that doesn't have an extra protective layer. Gram positives, the stain purple, that are more sensitive to penicillin, are sensitive to everything. Okay, the Lysols, the disinfectant cleaners, all of that. They're much more sensitive because they don't have an outer protective layer. All right. The gram negatives have a protective layer, and so they're tougher to kill. Now, if you add a protective layer that is made up of a whole bunch of lipids, oh my goodness. Now you're talking a very well protected cell. All right, it's not an endospore, but it's metabolically active. It's a real vegetative cell, but it is one of the toughest kinds of cells to kill because it has an outer layer of lipids. And this is what we see in the bacteria that cause tuberculosis, Mycobacterium tuberculosis. All right. To test for these kinds of bacteria with their really unusual outer lipidy protective layer, we do something called an acid-fast differential stain. It uses different chemicals and there are different color outcomes. So it's different than the gram stain. Different tests, different chemicals, different colors. Okay, but what we're looking for is this unusual protective waxy lipidy coat made up of mycolic acid, and it surrounds the cell walls of these kinds of bacteria. And this is Mycobacterium tuberculosis here. All right. So the type of bacteria that have mycolic acid are actually named after the mycolic acid substance. So Mycobacterium leprae that causes leprosy. Yeah, it's still a thing. Mycobacterium tuberculosis. Mycobacterium bovis. It's a kind of tuberculosis you can get from cow milk that you didn't pasteurize. Okay, so again, this is a different procedure. The acid-fast staining procedure starts off with a chemical that attaches strongly to mycolic acid. It is a red carbylfuchsin, and I know German terms. So you add the red color and it will attach to pretty much everything, but then you decolorize. Anything that is not acid fast turns blue, or well, anything that isn't acid fast turns clear, and then you have to add a counter stain in a different color. In this case, they chose something called methylene blue so that you can see everything else. But those organisms that have mycolic acid in their outer envelope, okay, they're going to be red carbylfuchsin. So acid fast is red, non-acid fast is blue. I can talk. So if you're looking at this, this is a lung that has been just bombarded and destroyed by a Mycobacterium tuberculosis infection. All right. So, how do you kill an acid fast bacterium that is so protected with that very tough, waxy, lipidy outer surface? Well, okay, it took a while, and it is Mycobacterium tuberculosis is technically also a gram-positive bacterium, but it has an outer protective lipidy area. So there are antibiotics that stop the production of mycolic acid, and there's a little enzyme, and there's a certain drug that will do that. And this is one of them. There's actually, when your patient is positive for tuberculosis, and you probably have run into this, they start off with 5 antibiotics, and then that switches to 3. One of the antibiotics tends to be isoniazid, abbreviated INH. So what it does is it takes out the enzyme that helps to make mycolic acid. If you really need to know, it's catalase peroxidase enzyme. So we use that again to treat mycolic acid containing bacteria like Mycobacterium tuberculosis, or if somebody does have Mycobacterium leprae, like leprosy. Like I said, it's still a thing. It's rare, but it's a thing. All right, let's switch gears for just a moment. All right, now if you're looking down at the picture, okay, we can look at the picture. They're really freaky, and they are. There are some bacteria that don't have a cell wall. So how do you classify that, Miss Eden? I'm glad you asked. Technically, they're still considered gram negatives, but they're not really, they're not the true gram negative types of bacteria. These organisms, these bacteria, have managed to survive without a tough, rigid cell wall by putting an extra little lipidy substance in their membrane, and this is what allows them to survive. They have kind of a special sterol that allows them to survive without a cell wall. It stabilizes them and makes sure that they don't explode. What kind of bacteria are we talking about here? Well, there's a whole group of these organisms called mycoplasmas, and they can cause anything from urinary tract infections to heart attacks. But probably the most common mycoplasma is Mycoplasma pneumonia. This one causes walking pneumonia. Yeah, walking pneumonia. So walking pneumonia, you've had this dry cough forever, and you just didn't get to the doctor. Maybe a low grade fever, but it wasn't bad enough to be seen clinically. So, all right, you kind of let it go, and then finally you go into the clinic, and they give you something that attacks cell walls, like a penicillin, and you take it for 7 days, nothing. Just doesn't even make, I mean, you might think it got better, but then it got worse. And yeah, it had really no impact on the disease and the course of the disease. Well, the reason is because there's no cell wall PBP enzyme. There's no cell wall, there's no PBP enzyme there. So penicillin's not going to have the slightest impact on these kinds of bacteria. So that these drugs won't have the slightest impact on walking pneumonia. Penicillins, drugs called cephalosporins, drugs, the carbapenems, they all attack PBP enzymes, and none of them will work on this because there's no cell wall. Okay. So again, they don't explode. They even look weird. They look like bacteria that don't have a cell wall. Like I mentioned before, the membrane, their one plasma membrane, have sterols, and that gives them strength and it keeps them from exploding. The reason we get pooped out with walking pneumonia is that they pretty much attach to our cells, and they're like energy vampires. Mycoplasma pneumonia. They attach by these little proteins, and the immune system doesn't do a really good job of seeing them because they have a way to just blend into our outer membrane and the chemicals of our membrane, but they are energy vampires. And that is why the cells just, they don't function very well. And sometimes they also cause our cells to form vacuoles, and cells can die that way. And yeah, we don't feel very good. So let's take a moment and review. What shape would you call these guys? That's one bacterial cell. Pleomorphic, many shapes. Yes. Okay, good. All right. So what is penicillin's target? The cell wall, the cell membrane, enzymes that make peptidoglycan, enzymes that make phospholipid bilayers, or DNA. Okay, if you said cell walls, you're wrong. Penicillin's real target is the enzyme that makes peptidoglycan. Okay, so it's the enzyme, and what do we call that enzyme? PBP, peptidoglycan building protein, or penicillin binding protein. Okay. But it's the enzyme that is the target. All right. Ultimately, that means the cell wall is not made well and it will explode due to cell lysis, but is the PBP enzyme. Okay. Why doesn't penicillin work for walking pneumonia? A, penicillin doesn't target bacterial structures. B, penicillin doesn't work on gram-positive bacteria. C, penicillin doesn't work on gram-negative bacteria. D, penicillin doesn't work on bacteria that lack a cell wall. E, all of the above. Well, this is Mycobacterium. Okay, so it doesn't have a cell wall. Penicillin isn't going to work on bacteria that lack a cell wall because these bacteria will also lack PBP enzymes. Right? All right. Based on this gram stain, what is this bacterial organism? Gram positive or gram negative? Okay, look at the dark places. This is a streptobacillus, by the way. Strepto arrangement, bacillus. Okay, shape. Looks like it's breaking down a little bit, probably because it's making endospores, but this is a gram positives. Stains purple, right? What about this one? This is E. coli. Yeah, this is gram negative. Very good. Based on the cell wall characteristics, which bacterial organism has more peptidoglycan in the cell wall? Gram positives or gram negatives? Gram positives. You got it. Okay, how about just once? Talk about a glitch. Based on cell wall characteristics, which bacterial organism would be harder to kill with a disinfectant and why? Gram negatives. Why? A freakish outer protective membrane. Okay, that would be true with antibiotics too. Okay, sheesh. Okay. Based on cell wall characteristics and cell envelope characteristics, which bacterial organism would contain an endotoxin that potentially produces fever and shock? Gram negatives. Right. And what do we call that? What's the endotoxin? LPS. Good. Gram positives, and remember, it's also antigenic. Gram positives have teichoic acid. Okay, which bacterial organism has a freakish double membrane unlike any other cell known on this planet? Gram negatives. Okay, what's a good example of a gram negative bacterial cell? E. coli. What's a good example of a gram positive bacterial cell? Staphylococcus aureus. Okay, staph. Okay. Based on bacterial morphology, which type of bacterium would typically be harder to kill with disinfectants and antibiotics? Gram positives like Staphylococcus and Streptococcus. Gram negatives like E. coli and Salmonella. Acid fast like Mycobacterium tuberculosis. You got that right. Yes, Mycobacterium tuberculosis. Absolutely. But what's the next one? Okay, well, oopsie. Sorry, the next one would be your gram negatives. Now, for those of you who know a little bit about Staphylococcus and Streptococcus, especially group A strep, you're probably thinking, now, wait a minute. I have a lot of patients that die from this and throughout the world. These are extremely medically significant. Yes, they are very common. They are very medically significant. I'm telling you, though, that on a counter, okay, he has some Lysol or spill a little bit of alcohol, it's going to be the gram positives that die first. Okay. And even with antibiotics, these are easier, not saying they're always going to work because there's resistant forms that we don't have antibiotics for. But for the most part, the gram positives are easier. Okay. But yes, they're very medically significant, absolutely. And they're more common. Okay, what about this one? What if I add endospores from Clostridium difficile? C. diff, right? Which one? Yeah, you got it. The endospores, remember, they're like immortal. If you can kill endospores from bacteria, bacterial endospores, everything else dies. Okay, we set the bar here. If it kills endospores, everything else dies. So this is why we wash our hands and reduce, 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 because you're not going to be able to destroy Clostridium difficile with Purell or anything like that. Okay. All right. Let's continue. Morphological characteristics of prokaryotic bacterial cells. Let's talk about the DNA. Remember, all cells have to have DNA, but prokaryotic, like bacteria cells, only have one single circular chromosome. And there it is. Okay, remember it's in the nucleoid area. Remember we call this the nucleoid area. There might be some plasmids there, but that's different. One single circular chromosome. All right, so remember, bacteria only have one single circular chromosome. All right, I got it. And it's in the nucleoid area here. All right. And again, there might be extra pieces of DNA called plasmids. That's what the arrow is supposed to point at. And again, plasmids can send genes from one bacterial cell to the next. You remember that, of course. When it comes to DNA replication, they have pretty similar enzymes that our cells have. They do kind of the same job, but they do have some subtle differences. And that is actually exploited. And some of these enzymes that work in bacterial DNA replication can be targets targets for antibiotics. So, yay, different bacterial enzymes are targets for antibiotics. And here's one, Cipro. Ciprofloxacin, levaquin, they belong to a class of drugs called fluoroquinolones. These drugs are going to target an enzyme involved in DNA replication. These are very powerful drugs, the fluoroquinolones, and should never be used lightly, okay? Because, okay, yes, they are going to attack an enzyme that is real similar to an enzyme our cells need, okay? Yeah, toxicity levels, again, there's a the therapeutic level and the toxicity levels are pretty close. Like the how much drug that you need to take to work can get kind of close to how much of the drug can cause toxicity. Okay. All right. So, fluoroquinolone drugs such as levofloxacin or levaquin and ciprofloxacin, Cipro, they are used for different kinds of bacteria. They are not used interchangeably. All right. And some of these are not used in young children because of possible toxic effects. But what do they do? Well, like I said, they are involved in blocking DNA replication or DNA synthesis. But what exactly? What's the target? Well, it turns out that bacteria have a special gyrase. Okay, it's a little, remember the enzyme that works in DNA replication, gyrase? Okay, bacteria have their own kind that is kind of different than ours. And it's supposed to keep replicating DNA molecules from breaking as it uncoils. So here we have gyrase, and as the superstructure, the DNA molecule uncoils, it's supposed to sort of uh, kind of break and reattach it so it doesn't split. But if this enzyme gets blocked, then what happens? If the enzyme gets blocked by a drug, then the uncoiling of the DNA causes the DNA structure to break. Now, that's a way to kill bacterial cells, right? So again, gyrase is used in whenever DNA becomes uncoiled. So it's going to be not just used for DNA replication, but also when you're going to make proteins. Remember transcription and translation to make proteins? The DNA has to be uncoiled to get a copy of that gene. So this is a very powerful drug. Unfortunately, it is misused quite a lot. And yes, it causes breaks in bacterial DNA. But it should never be used lightly. And is this going to cause cell death? Oh yeah. Yeah, like penicillin, it'll kill them. Okay. But again, human cells have a slightly different kind of gyrase. But we don't want to, we don't want to have to use that drug if we don't need to. Okay. What about those ribosomes? All cells have to have ribosomes because all cells have to make proteins. But as I mentioned before, prokaryotes have a uniquely small little ribosome. All right, so let's talk about if you remember your ribosomes and how DNA is put together, or excuse me, if you remember your ribosomes and how proteins are put together, ribosomes come in a large subunit and a small subunit, and they form around the messenger RNA. So here's our copy of our gene fresh from the DNA structure. And okay, so the ribosome starts right over the start codon. And okay, to make a protein, you have to bring in the amino acids. Well, what's going to bring the amino acids into the ribosome site? Well, that will be your transfer RNAs, and they come in and they drop off all of the amino acids, and the ribosome, of course, is going to link them all together. So what's the function of a ribosome? They make proteins. They assemble amino acids during gene translation. And again, ribosomes are made up of two subunits, which are different in bacteria. Bacteria have 70S ribosomes, and again, the S stands for Svedberg unit. And because this is an old way to measure ribosomes, the numbers don't add up. So the large subunit is a 50, the small subunit is 30, and so in bacteria, you add 50 and 30 as subunits to get a ribosome that is 70. It's that old way of measuring it doesn't make sense, but they just call the large subunits 50, the small one 30, and altogether it's a 30S ribosome. And there are certain drugs that attach to a 30S ribosome or a 50S portion of the ribosome, or some of them attach and block the transfer RNA or something similar. So again, this just shows here's the 30S and the 50S, and you get a 70S ribosome. I know, not my fault. Silly. Drugs that block protein translation. So these would be great drugs to use when you cannot use a penicillin. When would you not want to use a penicillin? Gram-negative bacteria. Yeah. Or bacteria that don't have cell walls, like mycoplasmas. Or if you're like me and you have an allergy to penicillins, yeah, I can't take those or some even similar ones. So then you go and you get drugs that block protein translation. Okay, so some of these drugs will bind to one part of the ribosome. For example, azithromycin. Okay, you know this is Z-Pak. Everybody loves the Z, don't they? Yeah, they love it because you kind of get it done, and compliance is pretty good with the Z-Pak, especially with little kids. But and relatively the side effects are rather low, and this attaches to the 50S portion of the ribosome. Okay, which is something that human cells don't really have, so it's not a problem. Tetracyclines, aka minocycline, doxycycline, these are going to block transfer RNA. Okay, so transfer RNA, what are they bringing in? Amino acids. So if you don't get amino acids in there, how are you going to make a protein? Oh, yeah, the cell can't actually. Consequences to blocking protein translation. No proteins for the bacterial cell. Now, here's one thing though. This is reversible. The effects of these drugs are reversible. You have to keep the concentrations up there. Why? Because if the bacterial cell has all of their ribosomes all blocked up, they'll just make more ribosomes. All right. So you have to so your patients have to be compliant. They have to keep the concentrations up there because this is temporary. This will not kill the bacterial cells. There are some protein synthesis inhibitors that do kill bacteria. We'll talk about those later. They're very toxic. But these drugs are not going to kill bacterial cells. They'll inhibit them, and hopefully they'll inhibit the bacterial cell until a white blood cell comes and gobbles them up. But until that happens, like 7 to 10 days, your patient has to keep the drugs in their system. They have to be compliant. Otherwise, the bacterial cells will just make more ribosomes that'll just make more proteins, and they're off and running once more. Okay. Let's talk about these neutrophils again. Oh, yes. So here's our white blood soldiers, and they have engulfed by phagocytosis these bacterial cells known as Rickettsia rickettsii. And remember, these cause Rocky Mountain spotted fever. And by the way, you can get that in this area, although it's very, very rare, but you can. You don't have to be in the Rocky Mountains. So, how can we send a poison down to hurt prokaryotic bacterial cells without hurting the eukaryotic human cell? Well, again, that drug better has to get in through our membrane, in through the bacterial cell membrane, and find something to latch onto that is bacterial and not human, okay? It has to attach to a unique structure, target a unique structure. So what have we talked about? PBP enzymes, although we tend not to use those for those drugs. But yeah, if they could attach to a PBP enzyme, that would be good. Or a drug that could attach to a ribosome or a part of a ribosome or transfer RNA. Or a drug that could attach to gyrase, right? To stop DNA replication. So that's what we do. All right. Yeah, that's what those would be. Which would be the safest antibiotic? Well, again, an antibiotic that attaches to something very unique in the bacteria. Well, okay, so our cells have ribosomes. And by the way, they're in our mitochondria. We even have 70S ribosomes. I know it's weird. If you want to know about it, email me. Um, we have a type of gyrase in our cells. So those can be kind of iffy targets. But if we could get a penicillin or a cell wall synthesis inhibitor, that is always the safest antibiotic. But to be honest, those antibiotics are too bulky, they'd never get in. So, but that is the safest antibiotic. Okay, so what process does Cipro inhibit? DNA synthesis, peptidoglycan wall synthesis, peptidoglycan cell membrane synthesis, or protein synthesis? Ciprofloxacin, levofloxacin, levaquin. Okay, yes, it attaches to gyrase, which blocks DNA synthesis. All right, it's not like penicillin that blocks peptidoglycan cell wall synthesis. And peptidoglycan is not part of the membrane synthesis, so that was misleading. All right, well, that's pretty dramatic, I know. Let's talk about bacterial toxins for a moment. All right. So, by the way, this is gangrene caused by a Clostridium, not Clostridium difficile. This one is Clostridium septicum. Now, like some other Clostridiums, it causes a gas gangrene. So you get the gas bubbles here as the bacteria ferment the tissue. Interestingly enough, this bacterium is also associated with some types of colorectal cancers. All right, so toxins. Here we go. So these are chemicals that bacteria manufacture. All right. And I don't know why, and here they are. So they can manufacture different toxins. So there are two kinds of bacterial toxins that we're going to talk about. And we put these into categories according to whether or not they are excreted into the environment or if they are part of the actual cell envelope. And okay, and they tend not to be excreted. Okay. So the first kind is an exotoxin, exo like exits. So the bacterial cells here are manufacturing the toxins and they are being released. This bacterial cell is making this exotoxin. This bacterial cell is making this exotoxin. Most exotoxins, but not always, most of them are from gram-positive bacteria. Examples for really some of the most powerful exotoxins come from Clostridium, like Clostridium difficile, C. diff, Clostridium tetani, tetanus, Clostridium perfringens, Clostridium botulinum, botulism. Also Staphylococcus aureus, staph. There are different kinds of staph. And strep. Streptococcus pyogenes, group A strep is the biggie there. Okay, so they tend to be really big at making exotoxins. These toxins are manufactured in the ribosomes and they're released outside, and they can go anywhere in the body, and they do. They do well, and one of the things about these toxins is that they are proteins. And as proteins, they can actually be seen as invading ID tags. They can be antigenic. So the body, if given time or a vaccine, can actually make antibodies against these. This is why we get our tetanus vaccine because Clostridium tetani makes toxins that will kill us. They will kill us unless we can get antibodies there. So antibodies are good at stopping these. So we can take preformed antibodies, sometimes known as gamma globulin. We can make our own antibodies if there's time, but if there's not time, we get preformed antibodies and we inject those into the patient to stop these toxins from going to their targets. The other type of toxin, and you've heard this one, this is the endotoxin. So endotoxins are they're part of the outer envelope of gram negatives, and they are only in gram negatives. Only. They tend to be released in greater numbers when the bacterial cells die. Some bacterial cells only release the endotoxins when they die. With Neisseria meningitidis, they can release the toxins in blebs as they replicate. So remember, the endotoxins are LPS, lipopolysaccharide, a lipid sugar. And again, it's in the outer freakish membrane of gram negatives. I told you there's a reason we want to learn about gram positives versus gram negatives. So again, in small amounts, fever; big amounts, shock and death in some cases. And they're found in true gram negatives and only in gram negatives. So would Clostridium or staph or strep make an endotoxin? No, they're gram positives. Okay? Gram positives never make an endotoxin. I hate to say it, but gram negatives not only contain endotoxins, but they can also sometimes make an exotoxin. More on that in just a little bit. All right. So exotoxins, as I said, they exit the cell. They are proteins, and they tend to be very specific with specific damaging effects. The bacteria don't have to be present anymore to cause disease. So even if you microwave a dish that's full of Staph aureus, the toxins may survive the heating process, and they can travel throughout the body or throughout the gut and cause problems. So exotoxins they are manufactured, they are secreted, they exit the cell. They can cause foodborne intoxication resulting from consumption. And again, about a third of, well, not again, but a third of the population carries staph. And even with the sneeze guard, sometimes things are not protected. So the action of exotoxins, well, it depends. Each exotoxin has a specific cell that it'll attach to. It attaches to receptors like hormones, if you remember that from your physiology. Okay, so it'll find a special little receptor on a special little cell. So if a bacterial cell secretes a neurotoxin, well, that is going to go and attach to a nerve cell receptor and damage a nerve cell and therefore all the cells in the nervous system. An enterotoxin, this is going to attach to intestinal cells and cause intestinal disturbances. So sometimes you'll see the word cytotoxin. This means that, okay, this is a general kind of toxin and it can attach to and cause damage to a variety of cell types. So if you're looking at this individual over here, and I know it's hard not to, I will tell you that Staphylococcus aureus makes an exotoxin that is also an enterotoxin. It's both. So think of Staph aureus in the food, manufacturing and secreting toxins. So you take this, and let's say you can take a doggy bag home and you put it in the microwave. Well, this particular toxin from this particular bacterial organism is very heat stable. So if you microwave the food, the bacterial cell will die, most probably, but the toxin will remain. Usually, when you eat food that has pre-made toxins in it, you're going to have a very fast, violent reaction. So with Staph aureus, you get what is called the double bucket flu, which I can personally tell you is no fun. I was at a Renaissance fair working, volunteering, and I had a rice dish because I love Indian food. So, yeah, I was really sorry. It took about 4 hours to kick in. If it's not the exotoxin that's doing it, it'll take a little longer. If it's viral, it can take even a few days. But yes, if it's bacterial toxin, it's going to be pretty fast. Sometimes it can be within an hour if it's a lot of toxins. Many of these toxins, these exotoxins, by the way, can be fatal before the immune response can even make protective antibodies. And like I said before, a good example of this is the exotoxin from Clostridium tetani that causes tetanus. Also, Clostridium botulinum that causes botulism. That is a deadly toxin. And again, the bacteria don't even have to be there. The toxin will remain. Do you think that these toxins are virulence factors? Oh, I would say yes. Anything that does damage to the host is a virulence factor. All right, so I wanted to all show you just to give you a reference, an idea as to how these exotoxins work and how specific they can be. So Corynebacterium diphtheria, we talked about this bacterial organism earlier. It causes the condition known as diphtheria. Thank goodness we have a vaccine. But here's the bacterial organism, and it manufactures this particular large protein and is secreted out. It is an exotoxin. The toxin will attach to receptors on cells. It turns out that Corynebacterium diphtheria is a general cytotoxin. It does like cells of the respiratory tract, but it will attach to many other kinds of cells. So it attaches to a receptor. The cell thinks that, oh, this is something that we're supposed to bring in by endocytosis. And it does, unfortunately. It's brought in by endocytosis into a vesicle. At this point, and a lot of toxins are in two subunits, an A and a B. There's a way to know the difference. The B tends to bind, and the A is the active portion that goes away. As the A portion of the toxin goes away, it causes changes within the cell. This particular one attacks our cell's ribosomes. So what does that mean? It means our cells cannot make proteins. What does that mean? That means our cells die. Okay? This is not a good toxin. Okay, there really aren't a lot of good bacterial toxins. But yeah, this one attacks our ribosomes. We're the ones that want to go out and attack bacterial ribosomes, but no, they come back and attack our cells' ribosomes and our cells' protein synthesis. This is not good. All right. So this is a reference just in case you have toxins that you want to talk about, an exotoxin that you want to talk about for your research paper. How about some other exotoxins? Well, some of them are very good at damaging blood cells, white blood cells and red blood cells. If the toxin is released and it goes after and punches holes in a red blood cell, then this toxin is known as a hemolysin. Hemolysin lyses red blood cells. There are many different kinds of hemolysins. Now, if the bacteria release a toxin hemolysin that causes the red blood cell to explode, and this is, I'm sorry, this is a sheep blood agar plate. It's red because it's loaded with red blood cells. As the bacteria were streaked onto the plate and they release the exotoxin and the red blood cells explode, you can literally read newsprint through what used to be blood in kind of a gelatinous agar. That's a beta hemolysis. There's also alpha hemolysis, where the red blood cells get holes in them. They get little pores, but it's a slow little leak, and the blood cells look the agar looks a little brownish, greenish. Now, this is really important, especially if you are classifying strep, group A strep, or one of the other types of strep. Because group A strep releases this kind of hemolysin. So, okay, so if group A strep, is growing on amongst red blood cells, it releases an exotoxin that causes the red blood cells to explode. And this is from nature, and it shows the red blood cell kind of falling apart here. So imagine the exotoxin coming out and forming pores in the poor little red blood cell. In the body, if you don't have blood cells carrying not carrying oxygen, sorry, you're going to be pretty weak. Your soldiers aren't even going to have enough oxygen to get ATP enough to make. So your soldiers are all going to be weak and you're going to have weak symptoms. Okay? So there you go. All right, some exotoxins can be super antigens. Now, what does that mean? Okay, I have the super here. This is a bad thing. All right. This is really, 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 really bad. So remember, antigen is anything that generates an immune response. What do you think if it's a super antigen? Oh, yeah. Most of the symptoms that your patients have due to an infectious organism is due not necessarily to the organism, it's probably not so much to the due to the bacteria, but to the inflammatory response that you get. And the inflammatory response, your body's immune system responding to the invasion, this is what gives you symptoms. Now, what happens if you have an overabundance of inflammation? So it's not just a battle in a few areas, it is an atomic war going on throughout the whole body. This is not good. So again, some exotoxins do freak out the immune response and become super antigens. So a super antigen is an antigen that activates a huge portion of the immune military force. It can stimulate anywhere from 5 to 30% of your soldiers, known as the T white blood cells, and these are the major soldiers in your immune response. Normally, an antigen only triggers about 0.001%. That's a normal antigen. This can go all the way up to 30%. You get something called an immune signal storm, known as a cytokine storm. Cytokines are immune signals, like hormones actually, but they're amongst immune cells. And the storm is so huge that can cause fever, shock, and sometimes a rash, and sometimes death. This is where we get toxic shock syndrome. Sometimes, and also something called necrotizing fasciitis. Some bacteria, Staphylococcus aureus, group A strep, releases antigens that cause such an inflammatory response that it will kill you. So Staphylococcus aureus and Streptococcus pyogenes. This is an example of necrotizing fasciitis, and this is due to all the toxins in the inflammatory response. This will kill you, right? Now, it turns out that group A strep, both group A strep and staph can cause this, but mostly group A strep. Um, they do make other toxins as well as the super antigen toxins, but this is not something you want to have happen, right? All right, what about another exotoxin? This one comes from botulism, or the organism's name is actually Clostridium botulinum. Now, when you have a patient that has classic symptoms of botulism, they will seem perhaps almost a little, so there's kind of that intoxication, the kind of the symptoms, the signs and symptoms. You get kind of this flaccid paralysis, meaning the peripheral nervous system is not working. It's like the outer, it's like the outer muscles aren't getting nerve signals. Okay. And that is true. It turns out that Clostridium botulinum, which is a gram-positive bacillus, releases an exotoxin that attaches to neurons, but the peripheral neurons, the neuromuscular peripheral neurons. So it's a neurotoxin, and it will cause paralysis, a flaccid paralysis. Flaccid is weak. Now, you can imagine your diaphragm is sort of around the periphery, right? And you need that to breathe. So this can kill you. This is a very nasty toxin. All right. So this is pretty much how it works. Now, hopefully you do remember from your anatomy and, well, actually it'd be physiology, but you have neurotransmitters, right? Acetylcholine is supposed to be released, right? And send a signal. Okay, I guess we have right here. So you have that released, and there's an enzyme called acetylcholinesterase, okay, that is going to be blocked by the toxin. And when that happens, you do not get the release of the acetylcholine neurotoxin. So the muscle cells aren't getting the sorry, the muscle cells are not getting the neurotransmitter acetylcholine, and therefore they don't function. You get paralysis. So the bacterial exotoxin, known as botulism toxin, attaches to the receptor on the motor neuron. So up here, the motor neuron, and it stops the release of the cell, or well, it stops the cell from releasing the vesicle full of acetylcholine, right? Because of that, there's no signal, and that means paralysis. So this is pretty much going to hit the peripheral nerves, and you get flaccid paralysis. Not good. Botulism is very serious, and I know we use it therapeutically, the toxin, but we really, really, really dilute it like it's an amazing dilution. And then it's very carefully administered, and it should be treated with respect. Okay, a very similar exotoxin comes from its cousin, Clostridium tetani. Again, it's an exotoxin, also a neurotoxin, but it's going to do something a little bit different. This is going to cause rigidity and spasms. So what this does is it does bind to the peripheral nerve, but but from there it will travel to the central nervous system. And if you're thinking, okay, this doesn't sound good, no, you're right. So what this does is instead of blocking the release of acetylcholine, it blocks the GABA. Okay, so it stops that neurotransmitter, and without GABA, well, okay, then you're not inhibiting your motor neurons. So this is why you get the severe contraction spasms and the rigidity. So the tetanus toxin does cause violent spasms. They will break bones, and they are quite deadly. Another reason why we like having our DTaP. We love our tetanus vaccine, right? We get that every 10 years because this is what happens. Yeah, this is not, it's not common. Tetanus is not common, but it is deadly. It is. The only thing you could do is you can try to block the toxin. You can give the patient preformed antibodies. That's the only thing you can do. And pray a lot, I suppose, to be sure you're going to need all the help you can get. But keep in mind, Clostridium, this is an endospore former. So this is blowing around in soil, out in the pastures. You don't need a rusty nail for this. Okay, all you need is a cut in your skin. The spore, the endospore, one of the toughest things on this planet to kill, the toughest thing on this planet to kill. And it gets into the wound, and from there on, okay, it germinates and it starts to release this exotoxin, and there you go. All right, not good. Okay, let's talk about E. coli again. E. coli is a gram-negative bacterium, so it does have that freakish outer membrane that has LPS. And that LPS, by the way, is very, 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 very heat stable. So heating it up is not going to work. Okay, but even more so than the last heat stable toxin we talked about. Interestingly enough, E. coli, if it gets a hold of a particular gene, it's a type of gene that it got from its friend Shigella. Um, it can also manufacture and release an exotoxin. And some of these exotoxins are heat stable and some of them are unstable. But the problem with this is if the E. coli that you consume in your food maybe it didn't get cooked, maybe it was a hamburger, it had a little fecal material in it, or the lettuce had some E. coli on it, you couldn't wash it off. It's in the gut, the bacteria are in the gut, but the toxins can leave the gut and go wherever. So, and the biggest problem is that these toxins that I especially refer to, known as Shiga toxin or Shiga-like toxins, they're exotoxins, and they will find your kidneys. The bacteria don't go to the kidneys, the toxins do. So, again, if E. coli gets this toxin gene, and it can get it through bacterial sex, we talked about, and other means. So, oops, the Shiga toxin that it can make is released. And by the way, E. coli isn't the only one that can make the Shiga toxin. There are some others that can do this too. but it releases the toxin. It can attach and attack the kidneys and cause something called hemolytic uremic syndrome. So the toxins are up in the kidneys, and this will also cause an inflammatory response. The inflammation is not going to be helped by antibiotics. Okay, I'll say this again. The inflammation is not going to be helped by the antibiotics. The antibiotics will kill the living E. coli cell. Okay, it'll stop the cell for it'll maybe cause the cell to die or stop it from making proteins, but the toxins are already there. And the toxins are chemicals, they're proteins, they're floating around and they're in the kidneys. All right. And they will cause an inflammatory response, just like I talked about with superantigens, but this is just antigens, period. And the inflammation will cause scar tissue, all right. And this can lead to maybe putting your patient on dialysis, maybe permanently. I had a student last year. She was all of 19 years old and she went on a picnic. They were rafting down in Valentine, Nebraska. And she had a little hamburger. And next thing she knew, she had E. coli, food poisoning, and it just happened to make the Shiga toxin. And she was on dialysis at the age of 19. Um, the reason I bring this up, not only is it something that you need to know, so again, the toxin attaches to these cells and it will stop the protein synthesis actually. So, of the cells that are here in the kidneys. But when you tell patients that they have to drink a lot of water, right? Let's say they have a bacterium like this, and the doctor is saying they have to drink a lot of water. It's not just because they're dehydrated due to diarrhea. Yeah, that's important. Absolutely. We don't want their blood going into sludge. That'll put them into shock. But the only way you're going to get the toxins out of the kidneys is by flushing those kidneys. And repeat after me, the solution to pollution is dilution. The solution to pollution is dilution. Get the patient drinking water. Flush the toxins out of the kidneys. You've got to get these out of here. All right. Yes, kill the bacteria, but get the toxins out because the antibiotics won't work on that. You've got to get them out there. Right? You don't want your poor patient ending up on dialysis. Like I said, in my student was 19, fresh out of high school. So, and here's a good example of how this particular Shiga toxin works. It attaches to a cell in one of the cells in your kidneys, and it's brought in by receptor mediated endocytosis, and it is processed in some of those membrane enclosed organelles we call the Golgi. And from there, it finds itself a ribosome, protein synthesis, right? And it blocks protein synthesis. And that's the end of your cell. And it'll also cause an inflammatory response, this toxin. The solution to pollution is dilution. All right, let's finish this up. LPS is a exotoxin, endotoxin, both exo and endotoxin. Exotoxin and antigenic structure on gram positives, endotoxin and antigenic structure on gram negatives. All right, this is kind of tricky. Think about it. Yes, LPS is an endotoxin. It's in that freakish outer membrane of gram negatives, but it's also antigenic, right? Yeah, so it's E. Okay. Oh, one last thing. Bacterial replication, I did want to mention this. It does occur by binary fission. It's a fast process, simple and fast. It's because bacteria are so simplistic. They don't have a lot of stuff to deal with. No mitosis, no meiosis. They have one single circular chromosome and some ribosomes. E. coli can replicate their DNA and their cells every 20 minutes under optimum conditions. So you get bacterial, like a doubling time or exponential growth. One cell becomes two in 20 minutes, two become four, four become eight. But that also means if you have 20 billion cells, you're going to have 40 billion cells in 20 minutes. So again, they don't have a lot to do When the cells are ready to divide, ribosomes gear up, make proteins. The single circular chromosome replicates. DNA is replicated. PBP enzymes make new peptidoglycan cell walls, shown here in orange. See, a PBP, if you block those PBP enzymes, this is all the parts of the cell wall that is weak. All right. One parent cell ends up making two identical clones we call daughter cells. And here we can see binary fission. And look how fast. This is exponential growth or doubling time. What about those eukaryotic cells, you ask? Okay, well, keep in mind, eukaryotic human cells, they're going to be different. Here's our white blood cell, our neutrophil. Let's blow this up a little bit. Okay, let's pretend this is our white blood cell, our immune cell in a body. Remember, there's a true nucleus here. You have multiple linear chromosomes. Just so number 2, right, is our true nucleus. Try to imagine the linear chromosomes. We have gyrase. It's a different kind of gyrase that helps DNA replicate. We have membrane enclosed organelles. So if there's anything inside, we got to get a drug that's going to go past our outer membrane and go get it. Okay. We have a rough endoplasmic reticulum with ribosomes. It's a factory because there's ribosomes here. And by the way, our ribosomes in our rough endoplasmic reticulum are 80S ribosomes. So they're bigger. They're bigger than what's in bacteria. So, all right. So hopefully they won't the drugs that bother 70S ribosomes won't bother our ribosomes there. But since these factories have ribosomes attached to them, they must make proteins. Yes, they do. Okay. You also have a smooth endoplasmic reticulum. And they're going to manufacture all kinds of lipid products, and they do a lot more. They process waste, detoxify stuff. Mitochondria, these are the powerhouses of the cells. They make ATP for the cell. When you eat food, most of it most of the energy and the food you take in you extract the energy here in the powerhouse of the cell. This is where you get ATP energy. So if your patient is anemic or not getting enough oxygen, let's say here's your white blood cell and your red blood cells aren't getting oxygen to it. Well, the oxygen is necessary to run the mitochondria. So if you don't have oxygen coming in, your mitochondria don't work well, you don't make a lot of ATP, and your whole cell is weak. And gosh, if this was a white blood cell, well, you don't want any of your cells weak, but yeah. Okay, so then there's structures called lysosomes. 10 and 11, well, we're just kind of doing 10 and 11. I don't know why that's off, but oh, I see. It's number 10, but structural, it's still weird. Okay, so you have these little pockets of these little membranous pockets called lysosomes, and they carry a powerful digesting enzyme called lysozyme, and this will actually break down bacteria. So it's kind of nice. Our cells will excrete this out into saliva and tears. So if bacteria try to get into our mouth or our tears, it'll help to break them down. But lysozyme only really works on gram positives. Yeah. So, and again, here's our white blood cell trying to phagocytize all the little bacteria. So little true bacterial or white blood cell, eukaryotic cell. One thing I wanted to mention here. So here we have Rickettsia rickettsii, and it doesn't get digested. There are bacteria that actually on purpose want to be eaten by some of our white blood cells. Why is that? Well, they've managed to block once they get phagocytized, they block lysosomes and therefore they never get digested. So let me get back to this thing. So remember the lysosomes? I know, pretend the green one's the lysosome. It has digestive enzymes. If the bacterial organism comes in, the lysosome fuses with the phagosome that carries the bacteria, and the enzyme should digest the bacterial organism. But what happens here is the bacteria have a way to block the lysosomes. So they get in and they can replicate, but they never get digested. And while they're there, they thrive. They live inside our cells. In fact, some of these, like Rickettsia rickettsii, that causes Rocky Mountain spotted fever, is an obligate intracellular parasite. They need to live inside our cells. They're obligate intracellular pathogens, and they thrive in here. And what a good strategy! Because would our white blood cells be able to find these guys? Not very well. So obligate means, yeah, and these guys, now there's a lot of bacteria that will live inside our cells. They're intracellular pathogens, but some of these are obligate, meaning they have to. Examples of obligate intracellular pathogens would be our Rickettsia rickettsii, causes Rocky Mountain spotted fever. You get a tick bite, right? And the tick spits out Rickettsia rickettsii into your bloodstream, and this is what happens. And they will also invade your endothelial cells, all right? And Mycobacterium tuberculosis, they get into macrophages and they can travel throughout the entire body that way, not just to the lungs. And of course, they're going to cause tuberculosis. Both of these are intracellular pathogens. Little side note about viruses here. Um, I just wanted to show you a virus. Here you go. You're like, what? Yep, that. Viruses are really bits of genetic information. When you see the pictures of viruses online or whatever, what you're seeing is the package that carries the genetic information. What the virus needs to do is they need to get the genes into our cells, and then they take over the cells. And using genes, using their DNA, they will manufacture more viruses. And that's really what a virus is. So viruses, are they living or are they not living? They're not self-replicating. They can't replicate themselves. They don't eat food. They don't respond to stimuli. They don't grow. They're actually manufactured when they're ready to go and infect another cell. Some could argue that viruses are maybe living once they take over your cell. So any cell that has a virus in it is no longer you, it's now a virus. But that's a weird kind of discussion with them. I I never thought of it that way, and I listen. My favorite podcaster is from Columbia University in New York City, Vincent Racaniello, and he has a wonderful podcast. He has several, but This Week in Virology is wonderful, and they argue whether or not a virus is living or not, and I can see both sides of the argument. But yeah, I mean they do. If they like HIV really does, it inserts its chromosome or it inserts its genes right into your chromosome. So it becomes your cell, and actually it's a white blood cell that it takes over. Hmm. All right. So what you typically think of as a virus is actually something called a virion. A virion is the infectious particle of the virus. So here the orange part would be the DNA or nucleic acid. What makes up a virion? Well, you have an outer protein coat, and I have that in green. Okay, so that's a coat or capsid. Viruses may not even have DNA, some do. Herpes viruses are DNA, but HIV is RNA, and the cold virus is RNA. So they have to have some nucleic acid, some genetic material. It depends. Okay. Now, if you put the nucleic acid and the capsid together, it's called a nucleocapsid. All right, and let me, oops, that is not spelled correctly. Sorry about that. All right, nucleic acid. Okay, now some viruses, not all, may have an envelope. Okay, and there it is in gray. All right, not all have that, but it's important. Okay, it may have something called spikes or papillomeres, kind of in orange. They may have some unique enzymes to help them grow. Sorry, they may have unique enzymes to help them grow in their invasion process. And hey, if they have unique enzymes, could we target those? Yeah, okay. But again, keep in mind, viruses don't metabolize, move, or grow. They are assembled, they have huge numbers, they bump into a host cell, they attach to the host cell, they invade the host cell, and they are assembled in the host cell. Okay, and they are very specific about which host cell they can infect. So, I'm sorry, they bump into the host, they grab onto host receptors, and the host thinks, oh, this is something good, just like with the toxins. And the host cell, like our cells, will bring in the viruses by endocytosis, just like our cells bring in bacterial toxins. It's crazy. All right, so here's our virus, here's our eukaryotic cell. All right, so in it comes by endocytosis. And all right, it doesn't exactly go straight in. There's some processes that it has to go through, but viruses take over our cells. All right, so here's a question. How do you kill a virus without killing the host cell? Find something unique in the virus. Exactly, you're learning. Good, good, good. If you can find something very unique in the virus, okay, then if you can target that, that'll target the viral uh, production, then you can do it. But you have to be very careful. I mean, the drug would have to enter the cell, and yeah, you have to know enough about the virus to know what might be unique. Usually, it's an enzyme. And that's usually the target if we can find it. There is another method. We can mutate all the DNA, knowing that the viral DNA will be replicating faster, therefore it will mutate faster. Um, but that's also going to mutate the host cell DNA. AZT is a drug that does that very thing. AZT is a mutator, it's a mutagen. And the idea is that it will hurt the viral replication more because the viruses are replicating DNA much faster. Okay. All right. All right. So, what do you call an extra round piece of bacterial DNA that are not chromosomal? Pili, fimbriae, plasmids, mesosomes, gyres, or transposons. Most of these we didn't even talk about. Okay, plasmids, not pili plasmids. It's the P word, but it's plasmids. All right. How do bacteria keep from being phagocytized, taken in by host white blood cells? And it's a virulence factor. Pili, capsules, plasmids, porins, or they just taste bad. Capsules. This is a streptococcal organism, and you can see the capsule in yellow. Hides them. How do gram-negative bacteria have XEX? Okay. Pili, fimbriae, flagella, pseudopods, cilia. Something called pili. All right. What would be the approximate size of a bacterial cell? 3 centimeters, 3 millimeters, 3 micrometers, 3 nanometers, 3 yards. Micrometers, right? You're in microbiology, micrometers. What is this bacterial shape and arrangement? Diplococcus, Diplobacillus, Streptococcus, Streptobacillus, Staphylococcus, Staphylobacillus, Streptospirillum, Staphylospirillum. By the way, spirili, the spirochete guys, they're always single. Okay, so none of those. This would be strepto, right? Bacillus. So we're in D. What process does azithromycin or Z-Pak inhibit? DNA synthesis, peptidoglycan cell wall synthesis, peptidoglycan membrane synthesis, protein synthesis. Protein synthesis, right. D, it attacks the ribosome directly. What process does penicillin inhibit? We're talking process. DNA synthesis, peptidoglycan cell wall synthesis, peptidoglycan cell membrane synthesis, protein synthesis. Peptidoglycan cell wall synthesis, yes. Which toxin is a protein that could be stopped by antibodies? Exotoxins, endotoxins, or both? Exotoxins. Okay, the endotoxin is LPS. It's a lipopolysaccharide. Okay. LPS is associated with gram positives, gram negatives, both or neither. Only gram negatives, B. Which antibiotic will work on this? Penicillin, tetracycline, Z-Pak, Cipro, Levaquin, none of the above. Yeah, this is a virus. Okay, it doesn't have a PBP, neither does its host. Tetracycline blocks transfer RNA, no. Z-Pak, ribosomes, nope. Cipro, gyrase, Levaquin, gyrase, yeah, none of them. F, none of the above. Okay, here's the joke. Two men walk into a bar. First one orders some H2O. The second one says, sounds good. I'll have some H2O too. Second man dies. What is that chemical? Yeah, that's hydrogen peroxide. All right, I know that was a long lecture, but I had an awful lot that I needed to cover, including I wanted to make sure that we had a lot of good questions in there. There's good topics. These are all things that we need to build on if we're going to continue the rest of the course, which of course we are. If you have any questions, make sure you give me a call. Email me, preferred method. And have a great week.